Good afternoon, ladies and gentlemen. Welcome the afternoon session of the second day of MESMAP symposium. Can you hear me?
I can hear you.
Okay, everybody. So we have two chairs for this session: Teresa Tutolomondo and Meryem Bozkurt. Teresa is here. Is Teresa? Yes. I will give you the chair, Meryem Hocam.
Good afternoon.
Hocam açılışı yapabilirsiniz. Şeyle beraber, Teresa ile beraber.
Hocam Teresa Hoca başlasın isterseniz.
Okay. Teresa.
Yes.
You can start the session.
Okay.
Each presentation 10 minutes. Thank you very much.
First presentation is adjuvant potential of plant extract in sheep vaccinated against Clostridia. Speaker is Sandro Carner Bana.
Marina, Sandra is here. Marina.
Can you hear me?
Yeah, who will present?
I will present. Dana, because of COVID, has lots of lessons, lots of teaching.
She's healthy. There is no problem.
No problem, but lots of
very
teaching.
good. Okay.
Okay, can you, I need to share my screen. It says that you
one minute.
Yeah, okay.
Yeah, now okay, try again.
Can you see my screen now?
Yeah. Full screen, please.
Okay. So please allow me to present some of our preliminary results on the adjuvant potential of some plant extracts, which is in the trend of our overall activity. Thank you very much once again for inviting our group of researchers, Nazim, and thank you for all the support to both you and Sevgi. The common control method for bacterial diseases and viral diseases in veterinary and both human medicine is the use of vaccines. This vaccination procedure is enhanced usually with chemical adjuvants. Due to the immune modulating properties and immune stimulating included properties of plants, our group is seeking to find some plant extracts which could increase the immune response to vaccines. We usually, within the framework of One Health, test plants which are already extracts prepared. We don't do our original extracts, and we buy them from regular natural pharmacies. So they are mainly for human use. So this would kind of provide us information also about the potential adjuvant use of these extracts for humans. This fragment of research aimed to investigate the immune enhancing activity of alcoholic plant extracts in vaccinated sheep. The experiment was carried out, as you see, in extensively raised sheep. The vaccine we used is one which is commonly used in sheep to prevent clostridial disease. At this point, unfortunately, Romania is under quite a big pressure of clostridial diseases. So this vaccine included, as you see, several strains of Clostridium against most of clostridial diseases of sheep which are observed in our country. This is an image where sheep are held during summer. In winter, they are usually introduced in shelters. The plants we used were several, known for their stimulating activities, anti-inflammatory activities, like Calendula officinalis, Echinacea, which is a very well-known immune-stimulating plant. Then we used Symphytum officinale, which is a common plant used in medicine to treat different diseases. So far, its immune-enhancing activities are not very well known. Then we used mint, which is again used for its anti-inflammatory activities, and we used birch extracts. Along with this, we tried to obtain some results for propolis, which is very well known for its antibacterial activity. And we did this because bee products are coming very much as therapeutical means, or spreading very much as therapeutical means in veterinary medicine as well as in human medicine. Blood from these animals was randomly sampled. We worked on a flock of sheep of over 200 animals, but we just sampled 10 of these. And we sampled blood before and 2 weeks after the vaccination, when immune stimulation is observable by tests available for this purpose. We tested the in vitro effects of these plant extracts, alcoholic extracts, by adding them to the blood, whole blood cultures, and compared with known immune stimulating compounds like phytohemagglutinin, LPS lipopolysaccharide, and concanavalin A. The plates were after the disposal, the sampling. We mixed the blood with RPMA culture medium for lymphocytes. We distributed this mixture in 96 well plates, as you see here, and then we added the extracts of plants and propolis, and also the standard nitrogens, and we incubated the plates for 63 hours at 37 degrees. And then we estimated the growth of the cells by looking at the initial glucose concentration of the culture medium and also the final glucose residue by orthotoluidine method. All the results were subject to statistical analysis, looking at the significance of the differences between the two samplings before and after the vaccination. Here we see the results in the control samples harvested before the vaccination. And what we can see in the activities of different extracts is that the Echinacea extract was well above all the others, stimulating the immune cells of non-sensitized animals. And it works even better than the standard immune stimulating compounds. As an overall percentage, the highest percentage was attained by the birch in concordance with propolis, sage, and symphytum. So this represented 31% of the overall activity of the plants. At the second sampling, very interestingly, after the vaccination and antigenic stimulation of the sheep, we can see that Echinacea dropped very much. Actually, it was the lowest stimulation index of all, while the birch extract had a very high activity. So what we could conclude from these results would be that the birch extract has a stimulating activity, and eventually, of course, subject to further tests, it could be used as an adjuvant to vaccines. Of course, comparing it with the alcohol, the solvent control was much higher, and then higher than all the standard nitrogens. Also, from the overall 26% of stimulation indices, the birch was the highest, 15%. So there were some results which benefited of statistical significance after 2 weeks of the vaccination in what concerns the effects of plants, tested plants. Echinacea had the maximal activity at the first sampling and the minimal activity after the vaccination, it stands for its immune modulating rather than stimulating effect. And looking at the before and after vaccination effect of individual plants, we can see that calendula dropped from 50 to 32%. Mint dropped from 54 to 30% compared to alcohol, which is the solvent. So very little, rather inhibiting activity of the mint. Echinacea dropped from 60 to 12%. This was the most dramatic drop. This is why I mentioned it could be immune modulating, reducing the immune response when it's too high. The birch had the best effect, increasing from 52 to 66%, statistically significant. And the propolis also, from its initial value, dropped to half. So what we could see as a conclusion of this study was that from all the plant selections and then this B product, the propolis we included, the birch extract was the best. And given further studies and further results, we could suggest this extract, alcoholic birch extract, as a potential adjuvant for veterinary vaccines. Thank you very much for your attention. If there are any questions?
Yes, I have one.
Thank you. I cannot hear you, I'm sorry.
Professor Nazlı.
Birch, okay. Birch extracts are known to have allergic activities also. Is there any risk for using them?
As I told you, we did not test them in vivo. It was a commercial extract which is used in treatment of common cold, usually. We just tested them in vitro. So the cells we stimulated here were mononuclear cells, were not basophiles, you know, and not mast cells. So eventually, as I said, subject to further studies to look more by other tests in the cell populations which are stimulated, like flow cytometry or something like that, we are planning to do further research. We could tell more about specific impact of this extract on certain cell populations.
Yeah, I see. But in any case, further studies performed on animals, in vivo tests on animals, and tested for their safety in regard to allergic effects.
So first step for us is always in vitro tests. Eventually, we go into cytotoxic effect of this extract to see the dosage. And then we eventually could try to do intradermal tests in non-vaccinated and vaccinated sheep to see if there is any local reaction, and then maybe mix the product with vaccine at the established dosage, and then see the immune stimulating effect by quantifying the antibodies against these vaccines.
Sure.
Because that would be the final stage.
Every compound, every substance may have some beneficial effects or some harmful effects, depending on their concentrations and their doses. Hope they help for sheep. Thank you very much.
Thank you very much for your question.
Meryem Hanım, anons edebilirsiniz Teresa'yı. Meryem Hanım.
I would like to invite another presentation, Teresa Tutu Lomondo.
Yes.
Are you ready? Professor Teresa.
Yes, yes. Good afternoon
Yes,
and
full screen,
welcome everybody to my presentation.
Full screen, please.
Can you see?
F5.
Listen.
We can put F5. Or put, okay.
Okay.
It's okay.
Okay. Thank you, Nassim, for the opportunity you gave me. Thank you, Mary. Well, the topic of my talk is the comparison of the current use of wild medicinal and aromatic plants for therapeutic purposes and for regional parks in Sicily. It is well now the large level of plant biodiversity existing in Sicily. Previous studies have, in fact, identified in the island more than 3,000 wild plants, of which 322 are endemic or sub-endemic. This high level of plant biodiversity has always been associated with the cultural traditions of the different Sicilian communities and in particular with food and medicinal uses. A number of studies highlight that wild aromatic plants in traditional medicinal preparations have always been considered by the local people as a rich source of ingredients capable of relieving pain and suffering. However, the knowledge of medicinal uses of those plants has to be correlated with the knowledge of the plant biodiversity of an area. So, the number and the quantity of species now in an area, the higher the probability of there being a high consensus factor of the medicinal uses of the plant by those populations. This study quantitative study report quantitative results of 2 years tests carried out in 4 regional parks in Sicily. The main aims of the study were to verify which wild medicinal and aromatic plants still used for medicinal purposes by the local populations and to analyze through quantitative indices the extent of the current knowledge of medicinal uses of these plants in the study area. In this study, we considered 4 mountain areas, which include most of the plant biodiversity of Sicily and are representative of the Mediterranean flora. They are the Madonie Regional Park in central Sicily, the Nebrodi Regional Park in northeastern Sicily, the Sicani Regional Park in western Sicily, and the Etna regional park to obtain information by local populations about the medicinal use of aromatic and medicinal plants. We conducted a large number of interviews in the 90 towns of the study area. In particular, the interviews selected elderly people who had spent their entire lives in the area and who had been traditional farmers. The plants collected were identified using a flora identification key. Subsequently, voucher specimen codes were created and specimen deposited at local herbarium in Palermo city. Five main indices were used for quantitative analysis of data. Cultural importance index, index of ethnobotanical diversity, ethnobotanical richness, evenness index, and informant consensus factor. In particular, that indices permitted to assess the richness of plant biodiversity in the study area, the number of useful medicinal species, how much consensus each species had within the local population. With regard to main result, a total of 802 people, of which 658 men and 144 women were interviewed. The informants were mainly elderly people who had spent their lives in the study area and practiced traditional farming, such as agriculture and livestock breeding. A total of 219 wild medicinal and aromatic plants were reported. They belonged to 62 botanical families. The most represented families were Asteraceae, Lamiaceae, and Apiaceae. Malva sylvestris was the species most cited by the Sicilian population. It is seen to help with digestive system disorders. A leaf decoction is used to treat gums and toothache, as also reported in other studies. Wool or crushed leaves of Clinopodium nepeta are generally appeared to want and have an homeostatic and cicatrizing effect. Decoction and infusion of seed and aerial parts of Feniculum vulgare are used to help digestion and in the treatment of gastrointestinal pain. Also confirmed by various regions in Italy and other Mediterranean countries. A considerable number of the species were used to treat one pathology only. Most of the species were used to cure the pathological diseases. Such as inflammation of the skin. Such predominance may also be connected to the agri-silvo herding activity in the study area. a high percentage of the species were instead used in the treatment of gastrointestinal system disease. The leaves and aerial parts of the plant were the parts most used in the phytotherapeutic preparations. The greater accessibility in ecosystems of the above ground parts of the plants may explain the high use frequency of that plant parts in traditional medicine. Decoction was the most commonly used preparation method followed by infusion. The finding of quantitative analysis show that Malva sylvestris, Clinopodium nepeta, and Laurus nobilis were the most important species to the study area in terms of medicinal use. This predominance can be explained by the fact that these three species are multipurpose, as they used also for food and aromatic purposes. Most of the species have, however, a very low cultural importance index, which would seem to show that little cultural importance is given to these species in traditional medicine in the study area. This could be seen as indicating that the local populations had little trust in some of the plants concerning the treatment of certain pathologies, or as a strong indication of a fall in traditional plant knowledge regarding medicinal use of plants. It is interesting to note that the average informant consensus factor value for all the illness categories was high, and these rivals high level of homogeneity in a consensus amongst informants in the study area regarding the medicinal use of set of species, and indicated that natural remedies are considered extremely effective. Concluding, it is important to observe that our results underline that current knowledge on the medicinal use of plants is not a predominant part of the culture of the local population of the study area. The percentage of species useful for their therapeutic purposes was found low in comparison with the floristic richness on the study area. The cultural importance index shows that of the most important species for medicinal uses are those with more than ounce use, mostly those which are also used for food purposes. Finally, there is a high consensus amongst the formants for only very few species, showing that natural remedies are considered to be effective only concerning few species. Thank you for your attention.
Thank you for your nice presentation. Is there any question? I think no question.
Okay, thank you.
Thank you very much.
I want to congratulate Dr. Teresa. It is really a good ethnobotanical study. I have seen a lot of Mediterranean plants. We have also in Turkey and Northern Cyprus. See you. Congratulations.
Thank you. Next presentation is chemical profile, antioxidant activity, and cytotoxic assessment of target recta. Speaker Anna Flavia Burklek. Are you? Burklek, Anna Flavia Burklek, and you? Are you? No? Next presentation. Comparative analysis of total phenolics, flavonoids, and antioxidant potential of methanol and acetone extract of sideritis nidiocomatosa. Speaker Sedzer. Elanur Simsek Sedzer.
Yes, I'm here.
Okay.
I think it's okay, but yes. Can you hear me?
Yes.
Yes.
We are sorry, there's an internet connection problem. Can you hear me? Can you hear me?
Nazım. Yes,
Yes.
Nazım.
Yes.
Okay,
Yes.
we are sorry, technical problem or internet connection. Now it's okay, I think. Our chairs, I think I will have a presentation. Our chairs, Teresa, Miriam.
Sì.
Can I start?
Allora, comparative analysis of total phenolics, flavonoids, and
Virgin olive oil.
Virgin
Olive
olive
oil.
a new novel source for omega 7 fatty acid.
1 minute, I can share my presentation and 5. Hem yönetmek hem şey gerçekten oldukça. 5. I think it's here.
Yes.
Okay. I will start now. So I want to mention about omega-7 fatty acids, novel fatty acids. This is, and we found the first time in the olive oil in our region. Yes, we have, we say that the origin of the olive is Kilis. Here is the northern part of Mesopotamia, very old region, and the humanity born from here, and go through to world. And we believe that olive also born here and go through the worldwide, Europe, America, Japan, all over the world. And in the historical research, 4,000 years seed kernels also found in our region. Kilisyalık olive varieties, formal variety, and grown in Kilis, Gaziantep, Şanlıurfa, Mardin. This area is southeastern part of Turkey and mainly Mesopotamia region. And this culture is a very important culture. So the highest olive ratio it has on the worldwide among the olive varieties in Europe, in other world varieties, olive varieties. Also, strong drought, resistance, durability to fall harvest damage, and resistance to pests and diseases, and suitability for regional condition and topographic structure is very important in for this olive culture. What we make in our study, we research phenological and pomological properties of the olive oil, Kilisyalık olive oil, in different locations and harvest time. We harvested the olive fruits from September to December 15. So every 15 days intervals. This is our old tree and its fruits for early harvest. And this is very special shape and high oil content, this olive fruits. And this is the early harvest olive oil. And this is my trademark, Altun Huzma olive oil, early harvest we produce here as monoculture, and last year we have a golden medal in Berlin, and Italy also high quality grades in olive contents. And in conclusion, we get different oil rates, olive oil rates from the different harvest time and location. We start around 8% olive oil in September, and then we reached around 32% olive oil in December harvest. And according to location also we get different ratios. And it was acidity. Free acidity is very important for olive oil, and it it means extra virgin olive oil up to 0.8. After 0.8, it's virgin olive oil up to 2%. And at the end of the last harvest, also we get virgin olive oil. We started extra virgin and we reached extra virgin olive oil in Kilisyalık olive trees, and it's suitable for Turkish food codex. And it's very important we found paulinic acid. It's omega-7 fatty acid, rarely found in the olive oil. Only I can reach only one word in a paper. There is no more scientific literature. Paulinic acid and olive oil. It's eicosanoic acid. Can you hear me?
Yes.
Yeah, I think there is some
connection.
Yeah. We are now very sorry about the connection problem. We know omega-3, omega-6, omega-9, and recently also we know omega-5 and now omega-7. Omega-7 fatty acids under the medicine companies for next plants, for the plants 2020 and 2028. They will work for omega-7 fatty acids. Why? These are unsaturated fatty acids and 7 carbon atom from empty carbon chain. The most common fatty acids are normally palmitoleic and gadoleic acid and vaccinic acid, but eicosinic acid, cis 13 paulinic acid is very rare and found in guarana. Guarana is very popular. Also, you can see from the television advertisement, this food additive or this cosmetic products have guarana seeds extract or guarana seed oil. So it's very useful for cosmetics due to their moisturizing properties. Also for diets, which omega-7 fatty acids have been shown to beneficial effect for HDL cholesterol and lower levels of the cholesterol. And it's encourage immune system. And paulinic acid around 7% in the guarana seeds, guarana fruit and inside the seeds like chestnut, and grown and used for some food additives and other. It's very important. Guarana have 4 times more caffeic acid than coffee. 4 times more caffeic acid than coffee. It's very important. And for cosmetic and care products, anti-cellulite creams and guarana shampoo is very popular nowadays. And in the near future, you will see more products from the guarana powder and guarana seed oil. In this study, unlike previous studies, paulinic acid, one of the omega fatty acids, were in the found olive oil, also known eicosinic acid, detected first time in our own olive culture, Kilisialı olive oil. And paulinic acid may be a distinguishing feature in early harvest, cold press, Kilisialı olive oil, and a novel data has been added to scientific literature. And also we can see this table from harvest times from September 15 to December 15, and paulinic acid around 0.40 around, and other fatty acids you can see here. Also palmitoleic acid, also omega-7 fatty acid, and gadoleic acid also could be found olive oil. We will work more for finding paulinic acid and palmitoleic acid and other omega-7 fatty acids in olive oil, Kilisialı olive oil. We want to thank you, the farmers, and it is difficult to grow olive and other products. So we are proud of health workers, but also farmers. It's very, very important for our next, our future, for our health. Thank you for your kind listening to me. If you have any question, I can answer.
Congratulations, my dear brother Nazım. It is really a good, innovative study to produce some products from the medicinal plants is very, very important for the countries. Thank you so much.
Thank you. Thank you. I am sorry. I am some excited. It's too difficult to manage and accept participants and give lecture and I'm sorry. But it's very important, and we will hear more omega-7 fatty acid in the next phase, and we will work more. Our olive oil is spicy taste, and also we get the prize in Italy. Rich aroma compounds. It's too difficult, Teresa.
Of course.
Teresa, do you agree with me, Cicely? I think, yeah, okay. And thank you. And another I will share after me. Cheers. Meryem Hanım, Teresa.
Next presentation, comparative analysis of total phenolics, flavonoid and antioxidant potential of methanol and acetone extract of Sideritis niviotomentosa. Speaker, Sedler.
Elanur.
Yes.
Elanur Şimşek Sezer.
Yes. I'm starting. Yes. Dear participants, my name is Elanur Şimşek Sezer. Can you hear me?
Evet.
There's not a problem, okay?
Yes.
My name is Salona Şimşek Sezer. I am from Selçuk University. Welcome to my presentation, comparative analysis of total phenolics, flavonoids, and antioxidant potential of methanol and acetone extracts of Sideritis niveo-tomentosa. The use of plants and their extracts for therapeutic purposes dates back to the ancient times and is the basis of many effective therapies. Plants are a source of a variety of natural compounds with different chemical structures and a broad range of biological activities. Plant biodiversity, with its richness and complexity, is the major source of the chemical diversity. Turkey, our country, is a key country for global biodiversity conservation. As a result of its location at the junction of three continents, as well as Anatolia's topography, complex topography, and geomorphology, Turkey hosts 11,707 plant taxa with 3,649 endemic species. The most active agents may apply to certain therapeutic areas are products of secondary metabolic pathways. Research in the field of biology, chemistry, medicine is directed at identifying and characterizing plant secondary metabolites with pharmacological activity that could be candidates for the synthesis of new drugs. The natural product database has a large number of unused scaffolds, and the differences between synthetic and natural products are remarkable. Among officinal plants, the Lamiaceae family includes some extremely interesting examples that offer natural substances and high-quality raw material. The family Lamiaceae consists of approximately 220 genera and 7,000 species worldwide. Some of the major genera belonging to the Lamiaceae family are Salvia, Manta, and Sideritis, respectively. Many species of the Lamiaceae family are considered to be in importance because of their use in medicine, cooking, and cosmetic industries. The genus Sideritis belonging to the Lamiaceae family includes herbaceous plants, commonly known as ironwood. Official species grow mainly in the Mediterranean, especially Spain, Italy, Albania, Bulgaria, Greece, and Turkey, where 140 native species are known, divided into roughly 320 subspecies, ecotypes, and cultures. Sideritis is represented by 53 taxa, and 40 of which are endemic in Turkey flora. All Sideritis species are known to be high in terpenes, flavonoids, and essential oils. Chemical components responsible for their pharmacological activity. Various studies explain the basis of the traditional medicinal uses of Sideritis species and sustain motivation to find new pharmacological effects. Moreover, a number of therapeutically useful compounds have been extracted from Sideritis species, and it was demonstrated that the genus is a valuable reservoir of bioactive substances. The therapeutic use of Sideritis species was first mentioned in Dioscorides in his book, De Materia Medica, written in the 1st century. Sideritis species have been traditionally used as teas, flowering agents for therapeutical purposes. The decoction or infusion, the aerial parts, orally or topically administered, are used in folk medicine as anti-inflammatory, anti-ulcerative, antimicrobial, antispasmodic, anticonvulsant, analgesic, and carminative agents. In Turkey, in our country, where Sideritis species are known as dağ çayı or yayla çayı, and Greece, the aerial parts of these plants are widely used to prepare herbal remedies and traditional teas. This tea, which is often served with honey and lemon, is well known for its pleasant aroma, spatial taste, and yellowish color. Sideritis tea is widely used against gastrointestinal disorders such as stomachache, indigestion, and flatulence, and in addition to alleviate the symptoms of common colds, including fever, flu, sore throat, and bronchitis, as well as tonic and diuretic remedies. Sideritis nivea tomentosa this studies plant is a local endemic species belonging to the genus, and there is a limited number of studies related to this species. Lastly, phenolic compounds are a main class of secondary metabolites in plants, and they are divided into phenolic acids and polyphenols. Many studies have reported the advantages of the phenolic compounds, such as anti-aging, anti-proliferative, antioxidant effects, and the increasing interest, the powerful biological activity of plant phenolics and flavonoids outlined the necessity of determining their content in medicinal herbs. Above all this information, the aim of this study to compare the phenolic, flavonoid content and antioxidant potentials of Sideritis nivea tomentosa extracts, which are prepared with different solvents. When we look at the material methods section, firstly, collection of plant material. The plant samples collected from their natural habitat, Antalya province, and they were dried in a suitable environment, suitable conditions, such as without sunlight, without moisture, and then powdered and extracted. By this way, 4 different extracts obtained from flowers and leaves of the plant by using methanol and acetone. The extracts coded as in this table. After the extracts were prepared, Folin assay was performed to determine the total phenolic content of the extracts. Aluminum chloride method for determine to their flavonoid content. And lastly, we used DPPH method to determine the free radical scavenging activities of the extracts. During evaluation of the data, we use different standards for each test. For example, for Folin gallic acid, for aluminum chloride assay, routine, and lastly, DPPH assay, we use ascorbic acid standards, respectively. Results and discussion part. When we talk about our Folin assay results, you can see the gallic acid graph of the foreign assay. The equation and R-square values are given. When we look at this table, the total phenolic content of the extracts were given gallic acid equivalent. We can talk about this table. We can clearly say that the methanolic extracts. Sorry, I can't hear. Total phenolic content and the methanolic extracts have much more phenolic content than acetone extracts. Total flavonoid assay, when we look at the total flavonoid assay results, you can see the routine standards graphic and the equation and R-square also given. When we look the routine equivalent table, we can clearly say that the leaf extracts leaf extracts have more flavonoid content than flower extracts. Lastly, the result of the DPPH assay in this graph, DPPH reduction capacities of the extracts are given. When we look at this graph, we can say that all of the extracts inhibits DPPH region as a dose-dependent manner. In this table, the IV50 doses of the extracts are given. According to this table, we can say that the methanolic extracts have lower IV50 values. This is meaning they have higher free radical scamming activity. Many studies have shown a positive correlation the phenolic compound content and the antioxidant potential of vegetables. Our results is consistent with this topic. In conclusion, Sideretis nivea tomentosa extracts, especially methanolic and the extracts prepared from leaves, are rich in phenolic and flavonoid content, and they have strong radical scamming effect. The findings from this study are promising for hereafter studies. This is a preliminary study for the biological activities of extracts. Our studies will continue for different properties of these extracts, such as cytotoxic, apoptotic, and enzyme inhibitory effects. These type studies, especially using extracts from the wild plant source, are very important in terms of more specific studies. Thanks for your attention and thanks for your patience.
Thank you. Nice presentation.
Sorry, may I ask some question, Teresa?
Yes.
Okay. Eleanor, first of all, I would like to congratulate you, but I have some question.
Yes.
You told me that Sideretis species you have worked on is a local endemic plants.
Yes.
Why did you collect these local endemic plants collected from nature? I know that Selçuk University has a big chance, has a big opportunity. Dr. Yüksel can cultivate all the medicinal plants in the garden of the agricultural faculty. I suggest you, especially for the endemic plants, do not collect from the nature. After cultivation, you may do your analysis, because for the environment, we have to protect our endemic plants.
Yes.
So how many grams did you collect Sideretis species from nature?
I can say that we collected small amounts with
1 kilogram, 2 kilogram?
Approximately 1 kilogram, but I don't remember, and low amounts. We can study especially low amounts of plants to the natural habitat threat. Not, I kept.
Of course, I understood you.
Tehlikeye sokmamak için, bitki popülasyonu tehlikeye sokmamak için muhtemel en az miktarlarda çalışmaya çalışıyoruz. Bunun dışında zaten hani bitkinin kesinlikle bir endemik bir bitki olduğu için bitkiyi kesinlikle riske atmıyoruz. Çok küçük miktarlarda çalışmaya çalışıyoruz diyebilirim.
Okay, you have collected a little amount.
Yes,
I
little,
understood.
very, very little.
Yes, my last question is, did you take any herbarium species number? Because you have to store the sample of the
Yes,
plants
I
in the herbarium.
A sample, but I forget in this presentation. My teacher, Professor Dr. Tuna Uysal, is collected this plant, and the collection number is his collection number, and the material was stored at the Kenya herbarium.
Okay, okay. Thank you. Good luck.
Thank you.
Next presentation, the karyotype analyzed in subtaxa of Centaurea pseudoscabiosa from Turkey. Speaker Miriam Bozkurt.
Thank you. It says thank you. The karyotype analysis in subtaxa of Centaurea pseudoscabiosa from Turkey, an important and relatively large genus in the flora of Turkey.
Elanur kapatabilir mi bilgisayarını?
Yes.
Kapattım hocam, kapattım.
Centaurea currently comprises almost 250 species distributed across Eurasia, especially in Irano-Turanian and Mediterranean regions. A broad redefinition of Centaurea has taken place in recent years, thanks to the generalized use of molecular, chirurgical, and biochemical methods. Comparison of DNA sequences finally allowed for delineation of monophyletic genus, Centaurea sensu stricto. This new definition, however, does not change the important fact that Turkey is the main center of Centaurea diversity. Centaurea is one of the large genus having highest rates of endemism in Turkey. According to latest reports, in Turkey, the genus is represented by 205 taxa, of which 115 are endemic, so that the endemism rate is about 56%. The largest section of Centaurea is section Acrocentron, which is still the subject of active research. The species of this section distinguished members of other sections within Centaurea by relatively large involucres, decurrent appendages, ending to robust spine and dimorphic acants. Turkey and the Eastern Mediterranean region have been suggested as of one of the oldest center of speciation of Acrocentron, mainly on a chirological basis. The section has two different basic chromosome number, 11 and 10. According to Garcia and Susanna, the incidence of count basic chromosome number 11 in the Eastern Mediterranean region is higher than in the Western area, even although recent survey have suggested that ratio basic chromosome number 11 per 10 is almost the same in both areas. The absolute number of species with basic chromosome number 11 in the Eastern Mediterranean region remains higher than in the Western Mediterranean center of speciation. Chromosomes usually give important information about plant taxonomy, particularly the number of chromosomes in each cell, size of chromosomes, centromere position, and behavior of chromosome are being studied. Even these chromosomal characteristics are important parameters for understanding intra-relationships and interrelationships of taxa. Many chromosomal and morphological studies have been carried out for the genus Centaura from Turkey and adjacent regions. One of these is Centaura pseudoscabiosa. There are chromosome counts in the literature related to this taxon. In addition, essential oil and secondary metabolite studies have been conducted on this taxon. And these compounds have shown significant cytotoxic and apoptotic effects on cancer cell lines. Additionally, this taxon is traditionally used in sickle ailments. The aim of this study to contribute to cardiological data for morphologically closely related Centaura pseudoscabiosa subspecies pseudoscabiosa, Centaura pseudoscabiosa varieties brokensis, and Centaura pseudoscabiosa subspecies glehni taxa. Material and methods. Plant material belonging to subtaxa of Centaura pseudoscabiosa were collected from localities of Turkey. Centaura pseudoscabiosa subspecies pseudoscabiosa Erzincan and Erzurum province. Centaura pseudoscabiosa varieties brokensis Gümüşhane province. Centaura pseudoscabiosa subspecies glehni Ardahan and Ağrı province. Collecting and choosing of mature seeds, taking of primer roots into hydroxyquinoline, treatment of carnor fixative, drying with acetorstain, observation with light macroscopy, karyotyping with cameram software, later chromosome reports. Results and discussions. The findings indicated that all taxa have diploid chromosome number. Our results show that the basic chromosome number was 11 in all population. The karyotyping analysis revealed the presence of metacentric and submetacentric chromosomes. Moreover, we determined the population taking place within subtaxa of Centaura pseudoscabiosa had both symmetrical chromosomes and specific karyotypes onto each subtaxa. Although all population of the subtaxa had relatively similar karyomorphology, they display clearly some differences in terms of chromosome size and variation as well as karyotype formula. The karyotype formula could be evaluated as very specific and fixed for population of each subtaxa, and it indicated that the subtaxa of the species were clearly separated from each other. Centaura pseudoscabiosa subspecies glehni had the highest total haploid chromosome length. Additionally, this subspecies had the highest the coefficient of variation of chromosome length, the coefficient of variation of centromeric index, and the mean centromeric asymmetry values among all population belonging to subtaxa of Centaura pseudoscabiosa. Centaura pseudoscabiosa, subspecies pseudoscabiosa, two populations were examined in this taxon. 11 pairs of metacentric chromosomes were observed at the mitotic metaphase. The taxon had diploid population. Centaura pseudoscabiosa variety spirocensis. At the mitotic metaphase, 10 pairs of metacentric and 1 pair of submetacentric were observed. The taxon had a diploid chromosome number. A satellite was detected in 1 chromosome pairs, and it positioned at the short arms of the 5th chromosome of this taxon. Centaura pseudoscabiosa, subspecies Glechny. 2 populations were examined. 8 pairs of metacentric and 3 pairs of submetacentric chromosomes were observed at the mitotic metaphase. The taxon had diploid populations. This table includes the chromosomal information of Centaura pseudoscabiosa taxa. The chromosome numbers and karyotype formula of Centaura pseudoscabiosa taxa are given in the table. The ratio of shortest and longest chromosome ranged from 1.54 micrometer in Centaura pseudoscabiosa subspecies pseudoscabiosa to 1.87 micrometer in Centaura pseudoscabiosa variety spirocensis. The smallest arm ratio was observed in Centaura pseudoscabiosa subspecies scabiosa, and the largest was observed in the Centaura pseudoscabiosa subspecies Glechny. The total haploid chromosome values show a moderately range from 16.86 to 26.13 among taxa. According to this table, the intrachromosomal asymmetry index varies from 80.49 to 16.92. The interchromosomal asymmetry index values found in the study ranged from 13.54 to 18.63, and the coefficient of variation of centromeric index varies from 3.87 to 10.5. Relationships among population of Centaura pseudoscabiosa based on the intrachromosomal asymmetry index and interchromosomal asymmetry index are shown in the figure. Both the intrachromosomal asymmetry index and the interchromosomal asymmetry index of Centaura pseudoscabiosa subspecies Glechny are higher than other taxa. Karyotype asymmetry indices have been widely used to make assumption about the mechanism of chromosomal evolution in plants. The coefficient of variation of chromosome length and mean centromeric asymmetry scatter pilots are preferred much more than scatter diagrams based on other incidents, because there are better suited to demonstrated karyotype relationships among taxa, especially when chromosome size variation is negligible. According to a two-dimensional graphic, Centaura pseudoscabiosa subspecies Glechny has the most asymmetric karyotype. There are previous records for Centaura pseudoscabiosa subspecies, subtaxa, with the same result. Despite the name and the morphological similarities, these results excludes any relationship to Centaura scabiosa with base chromosome number 10. Our result for Centaura pseudoscabiosa confirm the presence of basic chromosome number 11 among the Turkish Iranian species of section acrocentron. A previous hypothesis on the evolution of section acrocentron suggested that the Iranian group of taxa was derived, and thereafter no record the ancestral basic chromosome number was expected. In any case, this is only record of basic chromosome number 11 in area, as all the other Turkish Iranian species of acrocentron counted previously have basic chromosome number 10. This study determined the chromosome numbers, karyotypes, ideograms, and karyotype asymmetry degrees of 5 population in subtaxa of Centaura pseudoscabiosa. The karyotype of studied taxa were reported first time. In addition, karyological results supported that Centaura pseudoscabiosa varieties procansis, regarded as synonym in Flora Turkey, could be approached to revive in subspecies category. In conclusion, this study focused on chromosome number and morphology in subtaxa of Centaura pseudoscabiosa. Karyological studies within the genus Centaura of these karyomorphological findings will expect to make great contribution. Thank you so much for your interest.
Thank you, Miriam. Are there any questions or comments? No. Next presentation. Effects of fermentation on antioxidant activity of plant-based milk substitutes. Speaker Ajdar.
Elif Feyza Aydar burada mı?
Aydar.
Hello. Can you hear me?
Evet.
Sorry.
Elif Hanım, ekran paylaşır mısınız?
Can you hear me now?
Evet, duyuyoruz. Elif Hanım, duyuyoruz sizi.
Hello everyone. My name is Elif Feyza Aydar. I'm a master student at Istanbul Technical University Food Engineering Department. Firstly, I want to thank to all responsible organizers for this opportunity. Now, I will present you effects of fermentation on antioxidant activity of plant-based milk substitutes. It's a review study. The scope of this review study includes fermented foods and their health effects, non-dairy kefir-like fermented beverages, products in the global market, production of plant-based milk substitutes and fermentation, antioxidant activity, its definition, and some example studies on effects of fermentation on antioxidant activity of non-dairy kefir-like beverages. We will start with firstly fermented foods and their health effects. Recently, the demand on the functional foods is increasing and fermented foods are gaining popularity. They have been consumed since thousands of years ago. They are considered to promote health of their consumers. Fermented foods contain beneficial microbes, particularly lactic acid bacteria, and some strains of potential probiotics. They are responsible for texture, flavor, and better preservation of fermented foods. These bacteria have a significant impact on the well-being of humans. Health effects of fermented foods can be grouped into three, such as gastrointestinal health, immune health, and metabolic health. From the table, it can be seen that the health effects are really wide. They include infections, diarrhea, allergies, asthma, obesity, and diabetes. Plant-based diets is also gaining popularity because of numerous reasons. The most mentioned reasons are animal cruelty, a desire for a healthy lifestyle, and environmental awareness. Plant-based substitutes are popular beverages among vegans, but they are not consumed only vegans. Many people prefer to consume them to cow's milk. The reasons of this preference are lactose intolerance and allergy to cow's milk and health-related reasons. Plant-based milk substitutes are used also for essential ingredients to produce fermented plant-based foods for vegans, such as plant-based yogurt, cheese, kefir, butter, and ice cream. Although there are numerous brands of plant-based milk and yogurt substitutes, there are just a few of non-dairy kefir-like beverage brands, and they are produced from soy and coconut. However, there are several research in the literature about non-dairy kefir-like beverages. And now I have production steps. In this slide, you can see the detailed flow chart of plant-based milk substitutes production. The above part differs according to the raw material. Starting from the soaking in deionized water, the steps become shared. After soaking in water, draining follows. Then blanching, wet milling, filtration follow. Then homogenization, sterilization, and then fermentation begins. Firstly, especially for almonds, production starts with roasting step, which provides a neutral taste. Then peeling follows. It releases the toxic compounds caused by the shell, and bitterness problem is overcome. Then soaking in water follows. It provides swelling and softening, and this is a preparation step for extraction. Time needed for blanching decreases, extraction yield increases, off flavor reduces, and stability increases. Blanching decreases the microbial load, and enzyme inactivation is achieved. For example, lipoxygenase in soy is inactivated by blanching, which causes the beany flavor. In wet milling, raw material and the water are mixed, and then they are milled. Then filtration follows. It is applied to separate the cake and the milk part. Some emulsifiers such gum and gallium gum are used to enhance physical stability. Acid can be added to prevent oxidation. Some enzymes can be added and to increase the similarity with cow's milk, different raw materials. To increase the similarity with the cow's milk, we need to increase the protein amount in these beverages. For this purpose, different raw materials can be mixed. This also improves sensory properties. And also to increase vitamin and mineral content, calcium tristrate or numerous vitamins can be added. Homogenization pressure increases the stability, clarity, and whiteness index of the milk substitute, and then heat treatment follows. It is applied to extend shelf life and keep the quality high. So far, the plant-based milk substitute was produced. Then for kefir or any fermented food production, fermentation step begins. Fermentation of legumes, seeds, and nuts provide increased in their bioavailability. They are natural source of phytochemicals which have antioxidant effect, but those phytochemicals exist as bound and less bioavailable. Fermentation can alter this situation. Actually, fermentation of legumes, seeds, and nuts is common in Asian societies. From the figures, you can see some traditional foods from Asia. For example, tempeh from Indonesia, natto from Japan, and Chang Sheng from Korea. And now we will continue with antioxidant activity. In the left side, you can see the difference between a stable molecule and a free radical. The difference is an unpaired electron. The oxidative free radicals can be neutralized by enzymatic activity or by natural antioxidant. In the right side, you can see a free radical is becoming a stable molecule by the help of an antioxidant. Intake of antioxidant to the body is an important issue for the body health because free radicals can easily interact with cell membranes and with macromolecules such as lipids, DNA, and proteins. And they cause us disruption, and this leads to permanent damage and finally cell death. When enough amount of antioxidants are taken to the body, a balance between free radicals and antioxidants are provided. In the unbalanced situation, diseases occur. These diseases are oxidative stress-related diseases, including atherosclerosis, diabetes, chronic inflammation, neurodegenerative disorders, and certain types of cancer. Plant foods are natural sources of functional phytochemicals. Phytochemicals are non-nutritive, but they have protective or disease-preventive properties. Therefore, plant-based diets reduce the risk of oxidative stress-induced diseases. In addition to phytochemicals, amino acids, peptides, proteins, flavonoids, and other phenolic compounds found in the plant foods are also antioxidants, have also antioxidative effect, and they play a significant role as physiological and dietary antioxidants. Even though nuts, seeds, and legumes are rich in terms of antioxidant activity, plant-based mixed substitutes include smaller amounts of these beneficial bioactive compounds because firstly, they are lost during production. Secondly, the milk part only have hydrophilic phenolics, not hydrophilic phenolics. After filtration, the cake part and the aqueous part are separated. So in the aqueous part, we only have hydrophilic phenolics. For example, in sesame milk, only sesame lignans are found. According to the studies, antioxidants found in walnuts are lost during peeling, and less than 10% of antioxidants remain in the walnut milk. According to another study, the total phenolic compounds of hazelnut decrease about 42% when the hazelnut mix substitute is produced. Again, in another study, the total phenolic compounds of sesame decrease about 84% when the sesame milk is produced. Fermentation can increase the levels and bioavailability of phytochemicals as a result of enzymes and metabolites produced by the help of bacteria. For example, soybeans are unique among the legumes because they have high amount of isoflavones, which have high antioxidant activity. One of the studies mentioned about the transformation of isoflavone phytoestrogens to isoflavone applicons during soy milk fermentation. The concentration of isoflavone applicons increased 100%, and the amount of isoflavone glycosides reduced of 50 to 90%. The reason of these increases and decreases is beta-glucosidase activity. Beta-glucosidase activity is responsible for the conversion of isoflavone glycosides to applicon forms, and applicon forms are more bioavailable because they are more readily absorbed. Fermented foods, fermented non-dairy products are richer in terms of also mineral and vitamin content than plant-based milk substitutes. Calcium, magnesium, riboflavin, and niacin are found in higher amounts in fermented soy milk than unfermented soy milk. In addition, their anti-nutrient content are reduced, and by this way, they improve the immune system. Now, I will mention about some studies about effects of fermentation on antioxidant activity in non-dairy kefir-like beverages. The first study is about fermented hazelnut milk. In this study, after fermentation, phenolic content decreases. The reason of this decrease may be metabolization of phenolic compounds by kefir grains. The increase of radical scavenging activity is because of isoflavone-derived compounds. In another study, different ratios of coarse milk and soy milk were mixed, and their antioxidant capacities were compared. When we compare 100% soy milk and 100% cow's milk, you can see that soy milk kefir's antioxidant capacity is much higher. In the study below, is about soy milk fermentation. After 48 hours fermentation, 4% decrease in DPPH was observed, and total phenolic content was decreased about 77%. Again, this decrease can be because of Another study is about flaxseed kefir. Study all of antioxidant activity, phenolic content, and flavonoid content increased. The reason in the increase of phenolic content may be the linking of some phenolics that were bound to proteins, and increased antioxidant activity may be due to mobilization and production of individual phenolic compounds and proteolysis activity of the microflora. Lastly, in a study about peanut milk and skim milk mixture kefir, phenolic content, antioxidant activity, and lipid peroxidation activity increased. The reasons of these increases were told as the high level of polyphenols formed and peptides deriving from milk proteins generally. To conclude, non-dairy kefir-like fermented beverages have higher antioxidant activity due to formed phenolic compounds and bioactive peptides from proteins, and these beverages will continue to gain popularity because of their beneficial health effects. Thanks for your listening.
Thank you, Alex. Are there any questions or comments?
Dear Elif, according to your work, which kind of kefir should we drink? What is your idea? What is your suggestion to us? Did you hear me?
I cannot hear you.
We cannot hear.
Oh.
We hear. We hear.
Okay. Dear Elif, according to your work, which kind of kefir should we drink? What is your idea? Çalışmalarına göre hangi kefiri bize öneriyorsun?
I would actually prefer almond milk kefir, but it's not produced. Also in Turkey, there is no any brand producing non-dairy.
Okay, we should wait for the almond milk. Okay, thank you.
Yes, you can send us the samples, prototypes.
Yes, we should.
Evet, hocamla bana biraz şey gönderiyorsun.
Then you should send altın what's name? Altın üzme to us.
Tamam.
Of course.
Okay. Pleasure. I think it's okay. I would like to close the session. Thank you for chairs and speakers. It was a great session. And now some people may be tired and need 5 minutes, only 5 minutes break. And then we will start after 5 minutes, 2:40. Our chairs, 1 minute, yes. Our chairs are Kola Doikani and Filiz Meriçli, and we will start Kola Doikani. If everybody okay, we can start.
Thank you, Nazim.
Thank you, Teresa. And Kola and Filiz hocam.
Yes, we are ready, and bye.
We can start. Kola is ready, I think.
I'm here.
Kola, are you here?
Yes, you hear me?
Okay. We can start, Filiz hocam.
Okay. Good afternoon, my dear colleagues. Welcome to last session of the MESMAP 16. And
Bir tane daha var hocam maalesef.
öyle mi? Dünkü programda last'tı çünkü. Okay, I start again. Forget about the first sentences. Welcome to our session. I'm very happy to be with you. First of all, I would like to thank to my dear brother, Professor Nazım, and also his excellent team. And I would like to congratulate Sevgi for her new academic position. The first speech will be given by Kola Doğukani. She is also my friend as a chairwoman. Please, Kola, can you hear me?
Yes, yes.
It is a great pleasure for me to invite you.
Kola, you can share
Kola?
your presentation.
Yes, yes. It's okay.
Now we are listening you, Kola.
It's okay. The screen is okay.
No, not okay. You open your presentation and full screen, please. I can stop and again you can. Please share again.
Okay.
The first open your computer. Yeah, yeah, okay, I think.
And now it's okay.
Yes, we are listening you.
The screen is okay.
Yes.
First of all, hello, everybody, dear guests, participants, and organizers. I would like at first to thank or special thanks to Professor Nazım Tekiroglu and Dr. Sevgi Gizizi for giving always the opportunity to participate as a speaker, as a member in different committees and moderate in different sessions. It's very, very great and with pleasure that I'll collaborate and I work with them. It is not the first time. We are in 6th edition of MESMAP, so we are now friend of MESMAP. My presentation is entitled Assessment of Wound Healing Activity of Ethanolic Extract of Pistachia lentiscus, Leaves and Quercus ilex bark in full thickness skin excision in rat. This is work which collaborating with the Institute of Veterinary Science in my University of Tiaret in Algeria. As introduction, we should define some scientific aspects, like wound healing is a complex biological process in which the tissue repairs itself after injury. The healing process involves three overlapping phases, which are inflammation, proliferation, and maturation or remodeling. The preferable model to evaluate the effects of factors improving the wound healing process are excisional, incisional, and burn. We will see in our work that we are choosing excisional one. Several synthetic chemical products are used in wound healing, but they have major limitations such as complication and side effects. That's why we are choosing to work with herbal medicine that received a lot of attention in treatment of wounds and are widely described. Why? Because of their effectiveness, their reducing side effects, and their relatively low cost, their derivative and safe substances. In the Maghreb traditional medicine because I am from Algeria. Quercus electrux are one of the most commonly used plants to treat gastropathies such as diarrhea, gastric, and skin infection. For Pistachia lentiscus is the most commonly used in different regions, and its resin has been used for a variety of gastric diseases in the Mediterranean and Middle East countries, I think also in Turkey. The leaves of Pistachia lentiscus have been investigated as appetizer, astringent, to treat stomach acid, so a lot of biological activities. In addition, no previous studies were conducted to evaluate healing effects of local plants, such as pistachio and holm hock. Our objective is the evaluation of the wound healing effects of ethanolic extract of pistachio leaves and holm oak bark import thickness in Wistar race. So it is an in vivo study. For the material and methods, we are choosing to make this experimental protocol. First one is the choose of the plants. You are choose pistachio leaves and holm hog. We are making harvesting in terrace in the western north in the region of Algeria. We are making sorting, drying, grinding. This is for the preparation of our extract. We are making maceration using an as solvent ethanol 70%. For in vivo wound healing activity, we are preparing the ointment. I will say now how we are making this ointment. We're making cutaneous excision wound model, and for the evaluation of wound healing, we are taking in consideration the wound contraction percentage, the epithelialization period, and histopathological of skin. For animal, a total of 42 healthy adult male and female Wistar rates weighing 210 grams were purchased from Pasteur Institute in Algeria. Animals were housed in solid bottom polypropylene cages at the animal house of our institute during a period of 20 days for acclimatization before the experiment. The rats were maintained in lateral dark and light and at room temperature and approximately 55% of relative humidity. For the alimentation, they were provided ad libitum, and the research was conducted in accordance with internationally accepted principles for laboratory animal use and care in the European Community guidelines. The animals were divided into 7 experimental groups. There are 42. Group 1, negative control, they are left without treatment. The group B, positive control, they treated with ointment, which call it cicatril dew. The group C treated with pure. So I have a problem of sound. Maybe you mute the sound for participant, please. C treated with pure petroleum jelly. Group D treated topically with 10% of Quercus ilex. Group E treated with 30% Quercus ilex extract ointment. Group F and Group G treated with the Pistachia lentiscus with 2 concentration, 10% and 30%. For preparation of ointment, they are preparing like this: Pistachia lentiscus leaves extract, Quercus ilex bark extract, petroleum G to give a total weight of 40 and 40. For the cutaneous excision model, wound model, we are making a dorsal, in the dorsal region, we are making shaving 1 day before excision, and rates were anesthetized by intramuscular injection of ketamine. A full thickness, 1 circular excision wound at about 4 centimeters square were made using 2 coins and the sterile blade. Then rats were placed in individual and recent wounds. This was considered as 0 day. The next day, which is day 1, the rats were treated daily by local application of ointments according to their respective groups for 15 consecutive days. And the treatment consisted of full covering of the excision wound, which was lifted and rested. For the evaluation of wound healing, as I told before, we are looking for wound contraction percentage. epithelialization period, and histopathology of granular tissues. For wound contraction persistence, we are following the formula which is below. The epithelialization period was estimated, and the number of days required for the follow scab leaving no raw wound behind, and the following was taken at the end point of complete epithelialization. And for the histopathology of skin, at the end of the experiment, rats were sacrificed by deep ether anesthesia, and full skin sample were taken and fixed with 10% formalin. And after, we are making with the imatoxylin and eosin for coloration, and we are seeing under optical microscope. Second slide, we're observing for evaluation of histological changes, including collagen formation, presence of inflammatory cells, and so on. For the result, as you see in the table, we are making 4, 8, 12, 16, 20, 40, 24, and 28. And in the 24 day, we are looking at zero surface, so disappearance of the surface, and 100% of contraction. It means that there is a complete healing of the skin. For the period of epithelialization, we are reduction of the days, 14 for the control, but for the treated group, they are 10. This is for Pistacia lentiscus 10%, Pistacia lentiscus 30%, and for Quercus ilex 30%. But for Quercus ilex 10%, at 28, we have the complete healing in comparison with the control. This is figures which describe what I told now, and we are seeing the progression of the healing effect of of the skin treated by our extract. For the histological study, it is clear that we have the scare area which is rich in fibrous tissue and lacking skin appendage. This one compared to this which is normal, and this one thickening of the epidermis of the scare area. This is the formation of new blood vessel in the fibrous scare tissues orientated perpendicularly, and this is they show the absence of the muscular layer. This is the confirmation by the histological study that our extract are effective in wound healing of the skin. As conclusion, the ethanolic extract from Quercus ilex bark and Pistacia lentiscus leaf exhibited effective wound healing action in Wistar rat skin excision. Comparatively, both ethanolic extract-based ointment showing an accelerated healing process compared to rat from other groups. However, still be interesting to conduct further research for isolation, purification, and identification of the phytoconstituents involved in the healing activity of this extract. To the best of our knowledge, this is the first study of wound healing effect of ethanolic extract of pistachio leaves and holm oak, and now this article is under reviewing by an international reputed journal. Thank you for your attention.
Thank you so much.
If you have any question,
Yes.
oh, thank you.
Yes, please. What is the reason of using leaves, only leaves of pistachia?
The reason of?
Is there any specific reason of that?
Sorry, I did not hear you. Can you repeat, please?
Is there any reason for using leaves of pistachia? Why other parts?
Normally we eat pistachio fruit in baklava, in nuts. Why you use leaves?
Leaves? No, because for a scientific reason, one that we are looking that all the studies are about essential oils and so on. we are using the extract. This is the first point. The second point, that the leaves are not consumed. So we should valorize and exploit this real part.
Okay.
And the third one,
Okay,
that the leaves are rich in phytonutrients, because this study will be continued to see what are the chemical or phytochemicals existent in these leaves, which are responsible for wound healing effects.
Yeah, especially woods also are very active for their antioxidant activity.
Yes,
They
yes.
are very rich antioxidant flavonoids. Actually, we are currently working on other species of pistachia, Vera and Terebinthus in Turkey. So this plant is very fine to work on.
Yes, you know, but we in our region it is rich. That's why I chosen this in pistachio in the Tiaret region. It's very rich. That's why we are choosing to make experiment with the disponibility of raw material.
Yes, thank you very much for your
Not at all.
very nice presentation.
Do you
Thank
have
thank
pistachia terebinthus in your country? Terebinth.
I don't know. I will
small,
see.
small round fruits. Pistachia terebinthus. We have terebinth coffee, herbal coffee in Turkey.
Sure, in your market.
we need terebinth. If you have, we can import from Algeria 2 kilos.
Why not? We are always in collaboration, Professor Nazim. Dates, kernel, pistachio, and everything.
Thank you.
Thank you, Professor Nazim. But really, greetings, because we are feeling just that we are in Izmir. Everything is okay, chairman or chairwoman.
Thank you.
Chairwoman, great chairwoman.
Yes,
Great.
yes. Thank you, thank you.
Dear caller, congratulations for this excellent speech and presentation.
Thank you.
I think you are my friends for the
session.
Woman, yes, in this session. If you want, you may invite the next
yes
speaker.
Yes, okay. So now we have a presentation entitled Ethnobotanical Study of Medicinal Plants in Ain Antar Forest in northwestern of Algeria, which will be presented by Halal Nouria. So Halal Nouria, if you are here, please.
Halal Nouria, are you here?
I don't know. I'm seeing, but
Halal
I did not
Nouria.
find her. I did not find her name.
In the morning, she was here, but maybe
the
now,
problem of timing.
So we can use another,
Maybe.
and then we can. Abdul Qadir Rawas.
Abdul Qadir Rawas. I think he is
and
here.
Then, yes. And then
hello. So please, Abdul Qadir Rawas, give us your presentation, which is entitled Performulation Study of Nano Emulsion Preparation Based on Essential Oils for Wound Healing. So please, you have 10 to 50 minutes to give us the essential of your presentation. Thank you.
Thank you so much.
Full screen, please.
Hello, you are hearing my sound?
Yes.
Yes. Hello, everyone. My name is Abdul Qadir Rawas. I'm graduated master graduate from New University. My topic of study will be pre-formulation study on nano-emulsion preparation based on essential oils for wound healing. So the presentation outlines will be first the aim of the study, then small introduction, essential oils, especially oregano and rosemary essential oils. After that, a stage of these two oils. Then small talks about the system and emulsion system. After that, the conclusion. So in the beginning, the aim of our study was to formulate an emulsion formulation with essential oils as an active ingredient. يعني incorporation in the nanoemulsion system for the improvement and exacerbation of the wound healing process through inhibition of the growth of the bacteria in the wounds. Before that, we have to identify our problem. First of all, the wound healing is through 4 stages. The hemostasis phase, after the cut, the 5P and the P factor are working combined together to form the blood. After that, the inflammatory phase, which is the most critical and important phase among the other 4 phases, in which if we control this phase, the whole process of the wounds will be controlled. Then, يعني in macrophage and neutrophils will engulf the bacteria and scavenge the bacteria in this stage. So, for this reason, it's very important to control this phase, in which if you control it, the whole process, the other 2 phases will be controlled and speeded up. In a proliferative phase, keratinocytes and fibroblasts will work together to form the granulation tissue. After that, in which this granulation tissue will be replaced by collagen fiber to give the skin its normal shape. So, in acute wounds and chronic wounds, especially in chronic wounds, this our formulation will be so beneficial for them. As we know, for chronic wounds, the wounds will take much longer time to heal. So, if talk about essential oils. First of all, essential oils are volatile aromatic compounds extracted from the plants as secondary metabolites. They are composed of aldehydes, ketones, esters, ethers, and many other compounds. But mainly, the majority of the composition are terpenes, monoterpenes, sesquiterpenes, and others. The uses they are used since ancient time. They are used as preservation in the preservation of foods. They are used in medicine. They are used as spices. But in medicine, in folk medicine, they were used as anti-carcinogenics, prevention of atherosclerosis, anti-inflammatory, antiviral, and many other uses. So we talk among the other essential oils. We talk oregano and rosemary for their attractive antibacterial and antioxidant activity. For oregano bulgari species, oregano, it's considered in Turkey is the largest producers of oregano. Oregano is used until now. It's used as a spicy, and it's also in medicine used to decrease the level of low dose lipoprotein, the bad type of cholesterol. If we are talking about its role in wound healing, the obstacle of methicillin resistance to the focus areas was solved by oregano oil by its bacteriostatic and bactericidal activity. And also oregano showed in literature it destroyed the outer membrane of Pseudomonas aeruginosa by it has strong weakness of the microbes. If we destroy the membrane of the microbe, of course, it will give a weakness of the microbes. Talking about rosemary, the other Rosemary officinalis species, it was used to treat headache, to treat epilepsy, and to enhance the insufficient circulation. If we are talking about wound, it decreased the inflammatory cytokines that is related to the inflammation, and also improve angiogenesis, the formation of the new blood vessels, enhance the formation of granulation tissue, and the integrity of the skin will be restored. So we talk this antibacterial activity of, first of all, these 2 oils are extracted by gas chromatographic mass spectrometry instruments. A1 is for oregano and A2 was for rosemary. So we take analyze oregano, the main active constituent that was responsible for the antibacterial activity was carbachol with a percent 50.61%. And for rosemary, it was the main active constituent 1,8-CNO with a percent 39.35%. So for the antibacterial activity, the first test was proof macro-dilution test to test the bacterial minimum inhibitory concentration. We talk a bacteria that is the most one of the most associated bacteria in the wounds. That is the gram-negative Pseudomonas aeruginosa. The minimum inhibitory concentration for oregano was 0.77 microgram per milliliter, and for rosemary was 3.92. We are, as we can see here, the huge difference between them that shows oregano is much more effective than rosemary as MIC on the bacteria. So this was the first test. The second test was bacterial inhibition zone by disc diffusion method, also in the Pseudomonas aeruginosa. This one, A1, was for oregano. The minimum, the inhibition zone, the emitter was 10.5. I don't know if it's visible here, as we see here. This was for oregano, and this was for rosemary. So the difference between them are obvious. So, as a pharmaceutical technology department, we need to incorporate our active ingredient in a system. So we put in a nanotechnology system, nano-emulsion system. It's very useful system due to its normal small size. It's a hydrocolloidal system made up of two phases: the oil phase and aqueous phase. These two phases are stabilized by the aid of surfactants. This is the surfactant, the hydrophilic and hydrophobic tail. So the components of our formulation were oregano oil as active ingredient and as oil phase, and polyphor RH40 as a surfactant to stabilize the system, and purified water was as an aqueous phase. So the most important characterization of nano-emulsion are droplet size, zeta potential, and polydispersity index. These three characterizations, our optimal formulation with these results, the droplet size was 16.94 nanometer, and the zeta potential minus 6.79, and the PPI was 0.208. This is very optimum and excellent result of the formulation. As a conclusion, the obtained results from the show results of oregano and rosemary, the results show that oregano is much more effective than rosemary. This is the oil, not the formulation. So we will ensure that this study is the preliminary study for our formulation. So according to these excellent results, the bacterial growth in the wounds will be inhibited, leading to speeding up the inflammatory phase. As we say, this is the most important phase among the other phases, leading to speeding up the inflammatory phase of the healing process. Hence, the overall health of the skin will be restored faster. I'd like to acknowledge my advisor, Associate Professor Dr. Yabi Souza, and for the excellent team together. And also, I would like to thank Professor Dr. Nazim Shekil and for the Associate Professor Dr. Sergi Geziji for organizing this conference and offering me the chance to participate in it. And thank you.
Congratulations, Abdul Kader. I am proud of you because your advisor is my student from Ankara University, and you work at
This is my honor, actually.
I'm proud of you. Is there any question? Everything is clear, I think. Kaola, may I introduce the next speakers?
Yes.
Okay.
Yes.
Now, Etil Meriç will present her work titled Standardization Studies on Silver Lime, Tilia tomentosa, used as a traditional medicine in Turkey. We are listening you, Etil.
Thank you so much, first of all.
You're welcome.
I think it is a full screen now. Is it okay?
Yes.
Okay.
Thank you so much. First of all, I would like to thank you all for being here, and special thanks to organizing and scientific committee for such great organization. Today, my topic is standardization studies on silver lime used as a traditional medicine in Turkey. I will talk about general information of Tilia species, the aim of the study, and then methods used for standardization of the silver lime inflorescence. Then I will talk about the results. Lime trees, which are also known as linden trees, may reach up to 40 meters, and their yellowish-white colored flowers have a nice scent. Tilia cordata, Tilia platyphilos, and their naturally occurred hybrid Tilia vulgaris are registered to European pharmacopoeia. However, these species are rare or absent in Turkey. On the other hand, Tilia tomentosa, or as known as Tilia argentea, is widespread in Turkey, especially in Marmara region. It is also known as silver lime due to the silvery, as you can see here, silvery color of the lower surface of the leaves. That is because dense stellate trichomes accumulation. Now, I would like to talk about chemical composition and bioactivity of inflorescence of Tilia species. It has campherol derivatives, one of which is tilirosite, was first isolated from the leaves of Tilia tomentosa. Hyperosite, isoquercitrin are from quercetin derivatives, and all these mentioned compounds, tilirosite, astragalin, hyperosite, isoquercitrin, have anti-inflammatory, anti-proliferative, antioxidant, immune-modulating activities. When we look at the caffeic acid derivatives, caffeic acid, chlorogenic acid have antioxidant, antimicrobial effects. And polysaccharide fraction, they have antitussive and immunomodulating activity. Lastly, its volatile oil has an antimicrobial activity. Due to the wide range of health benefits, Tilia tomentosa containing herbal supplements are commercially available on the market. Here you can see some examples. Due to not having the standard procedure in the pharmacopoeia, it's not possible to talk about quality control mechanism in place. So there is a need of herbal drug standardization. Therefore, we focus to compare the characteristic properties of official species with Tilia tomentosa within the scope of preparation of a monograph for Turkish pharmacopoeia. Now, I will mention about the methods used in standardization of silver lime inflorescence. We use European pharmacopoeia as a reference book. We performed macroscopic and microscopic analysis, high-performance thin layer chromatography, foreign matter, loss on drying, total ash, and additionally, we performed swelling index text and high-performance liquid chromatography. Here you see the samples. We used 12 Tilia tomentosa samples collected from different regions in Marmara. And for comparative analysis, we also collect Tilia cordata and Tilia platyphilus. Based on macroscopic analysis, Tilia tomentosa bracts are ribbon-shaped and they have hairy texture. The number of flowers can reach up to 10. Bracts, together with flowers, forms the inflorescence. When we have a look at the leaves, they have a cordate shape and lower parts silvery due to the stellate trichome. Here you see the microscopic analysis. We focused on bracts and different parts of flowers. The only difference between official types and Tilia tomentosa is the number of branches of the stellate trichomes in ovary. Official ones have 2 to 4 branches. On the other hand, Tilia tomentosa has 4 to 8 branches. In HPTLC method, we use silica gel as a stationary phase and the mixture of ethyl acetate, formic acid, and water used as a mobile phase. After development, first we derivatized the plates and documented them at 366 nanometers. Here you see the sample and standard preparation. Rutin, chlorogenic acid, hyperosite, isoquercitrin, astragalin, quercitrin, tilirosite, and caffeic acid used as a standard mixture, and hydroalcoholic extract used as a sample. Here you see the fingerprints of different parts belong to Tilia cordata and Tilia platyphilus. All investigated compounds, except rutin, was found in these samples. Now you see the fingerprints of other samples. First one represents the mixture of Tilia cordata, Tilia platyphilus, and Tilia vulgaris, and the rest represent Tilia tomentosa samples. Differently from Tilia tomentosa, we found rutin. As I mentioned before, we couldn't determine rutin in Tilia cordata and in Tilia platyphilus, so we believe that rutin comes from Tilia vulgaris. Accordingly, with European pharmacopoeia, foreign matter should be maximum 2%. And in our investigated samples, all samples met the criteria. One of the other parameters is the water content. So we applied loss on drying test. And the maximum value, again with according to European Pharmacopoeia, the maximum value should be 12%. All samples met the criteria. To determine inorganic impurities, we applied total age. And accordingly with European Pharmacopoeia, the maximum value should be 8. 2 of the tested samples were not met the criteria. Swelling index gives an idea about the mucilage content of the drug. This test hasn't been taken into consideration in the European Pharmacopoeia. As it's a new parameter, we need to define a value for the evaluation of the drug. So we took the average of the tested samples. Accordingly, swelling index value should be minimum 5. Lastly, a newly developed HPLC method for quantification of isocalcitrin and tildroside was validated for specificity, linearity, limit of detection and quantification, precision, and recovery. We follow the same procedure as before. When we averaged the results, we found that minimum content for isocalcitrin should be 6 mg per gram, and for tildroside, minimum content should be 2.5 mg per gram. As a result, our study will be published as a monograph of a silver line inflorescence in the Turkish Pharmacopoeia. Thank you for listening.
Thank you so much, Etil. It was really good presentation. Is there any question, our dear colleagues? No question.
Very clear.
Thank you so much.
You're welcome. Really good presentation. Everything is very clear, I think. Many greetings to Yeditepe Pharmacy and Marmara University Faculty of Pharmacy. You work together, I think, with Gizem.
Yes, we are collaborative working with Marmara University and other universities also.
I'm very happy that the young scientists collaborate with each other. It is really good for the future. Congratulations.
Thank you so much. Nice to meet with you. Thank you so much.
You're welcome.
And now, yes. And now, please, we have Najat Agiel, I think.
Yes.
From Northern Cyprus. Your title of your presentation is Prospective Crataria Species from Libya and Northern Cyprus for Production of Herbal Medicine. So please, you have from 10 to 15 minutes for the presentation.
Thank you.
That's all. Okay.
Najat Agiel.
Okay, can you see my screen?
We are waiting. Yes. It is ready.
Yes.
Full screen, please.
Full.
Okay. Hello, everybody. First of all, I want to thank Professor Nazim for organizing this MESMAP 2020 in this difficult, hard times. And I want to present part of my PhD study, which includes the prospective Crataegus species from Libya and from Northern Cyprus for the production of herbal medicine. My presentation will include the introduction and the aim of the study, the plant material that has been used, phenolic compounds extraction and HPLC analysis, and I will end up with the conclusion. Nowadays, we know that cardiovascular diseases are considered a serious health problem, and the mortality rate is increasing every year according to the WHO website. On the other hand, ethnobotany documents that the Crataegus species are one of the most outstanding medicinal plants in the treatment of the cardiovascular system. Scientific research also documents the effectiveness of the Crataegus species in the treatment of circulatory problems. And we know that therapeutic preparations from the leaves, flowers, and the immature fruits of Crataegus species has been used in the treatment of the cardiovascular system disorders since the 1st century AD. Now, since the interest in the herbal medicine has been growing, many pharmaceutical companies are commercializing herbal drugs. And today, we can see a number of herbal medicinal preparations from the Crataegus in different dosage forms that are present in the pharmacies, like teas, tablets, syrups, tinctures, and oral drops. The biological active metabolites that are responsible for the activity of the Crataegus species are the flavonoids and the glumaric procyanidins. And the extracts prepared from the Crataegus has already been documented in the pharmacopoeias as commercial extracts of standardized composition and for the production of herbal drugs, whereas many other Crataegus species has not been investigated so far. So the aim of our study was to evaluate the possibility of 2 Crataegus species for the production of Crataegus-derived pharmaceutical products and meet the requirements of the pharmacopoeias. For this purpose, an analytical method was developed for the identification and determination of the most important bioactive flavonoids. The plant material that has been used, as I said before, is 2 Crataegus species, one from Libya, Crataegus pallasei, and Crataegus azoralis from northern Cyprus. And for comparative purposes, Crataegotropophin, the Crataegus-derived herbal drugs, was used. Crataegus pallasei, it grows in the city of Ilmerj in Libya. This city is situated at the northern eastern part of Libya. It lies in an upland valley, which is separated from the Mediterranean Sea by a range of hills that comes from the Jebel Akhdar mountains, eastern to Benghazi, the 2nd largest city of Libya. Crataegus azoralis samples were collected from Genghis Khoi in Lefka in northern Cyprus. Crataegus-derived phytopharmaceutical drops, Crataegotropophin, was purchased from Maritim Apotheke in Germany in 2018. The phenolic compounds extraction and the HPLC analysis, and I should proceed first with the extraction process. The dried powder of Crataegus leaves and flowers were first extracted in a Soxhlet apparatus using ethanol to obtain the ethanolic extract. The ethanolic extract was further fractionated to obtain the phenolic-enriched extract ethyl acetate, which was then used for HPLC analysis. And for this purpose, the standard Vitexine 2-O rhamnoside, rutein, Vitexine, and hiproside were used. However, the initial chromatograms, they showed elevated baselines due to matrix effect, which poses a risk for error in quantitation, especially when the analytes are present at low concentration near the limit of quantitation. So therefore, indirect dispersive liquid-liquid microextraction was developed to overcome this problem. In the conventional dispersive liquid-liquid microextraction, the analytes are usually extracted into the organic phase, where they are pre-concentrated, and the analyte-rich extraction phase is separated from the mixture, evaporated to dryness, and the analytes are reconstituted into a water-miscible solvent for HPLC analysis. In the indirect dispersive liquid-liquid microextraction technique, the potential matrix constituents which would interfere with the analytical peaks were extracted into the organic phase, which is chloroform, while the analytes remained into the aqueous phase, which was there later injected into the HPLC for analysis. Therefore, for our sample preparation, we used indirect dispersive liquid-liquid microextraction. Our sample in the aqueous phase was injected into acetonitrile and chloroform as a dispersive and extracting agent, vortex, centrifuge, and the analytes or the compounds were in the aqueous phase, which was later injected into the HPLC for analysis to obtain our results. Here we can see the difference before and after this indirect dispersive liquid-liquid microextraction in the baseline. Optimization for the chromatographic condition was carried out by one factor at a time approach. And for this purpose, and relying on the resolution, separation, and peak efficiency, we used acetonitrile, was better as a mobile phase. And also the final and the initial concentration of acetonitrile was 60% and 20%. The gradient time was chosen to be 12 minutes, and the temperature was taken to be 20 degrees centigrade, and the flow rate was taken to be 0.8 milligram per minute. The our method shows, according to these figures, good linearity, as the coefficient of determination was more than 0.9950. It also shows good sensitivity, according to these figures, and good precision, and also good accuracy, as the relative recovery of the analytes was more than 98%. From the chromatograms, 4 peaks were identified, which were vitexine 2-or amnoside, butene, vitexine, and hyperoside. And from our results, we can see that the percentage concentration of the compounds in Crataegus azoralis and Crataegus palaceae, showing that hyperoside was higher concentration in Crataegus palaceae, whereas vitexine 2-or amnoside was in the same concentration for Crataegus azoralis and Crataegus palaceae. Again, Crataegus azoralis shows the same concentration for vitexine 2-or amnoside and hyperoside. According to the United States Pharmacopoeia monographs, the standardized extract of Crataegus leaf and flower is required to contain not less than 0.6% of the C-glycosylated flavones expressed as vitexine, and not less than 0.45% of the O-glycosylated flavones expressed as hyperoside. Whereas on the other hand, the European Pharmacopoeia states that the standardized hawthorn leaf and flower extract is required to contain at least 0.8% to 3% of the total flavonoids based on hyperoside. From the requirements that are stated in the international pharmacopoeias, and according to our results of the HPLC analysis study, and in comparison with the Crataegus-derived Crataegus tropophin, we can say that the Libyan Crataegus palaceae and the Cypriot Crataegus azoralis are proposed as a novel plant crude natural material for the production of Crataegus phytopharmaceuticals. These are some of the photos in MESMAP 2017 with my professor, and also in WOCPAP 2019. Thank you for your attention.
Kohala, please open your microphone.
Sorry. So thank you, Najat Aguirre, for your nice presentation.
Thank you.
Congratulations and best wishes and good luck for your PhD students. I have one question, please. If I don't know if I am understanding or not, why you are choosing Crataegus tropophin?
Crataegus actually is a widely used Crataegus-derived herbal drugs. It is used commercially to be sold in the pharmacies. It is a well-known drug of Crataegus. That is why we chose Crataegus.
Just for comparison, to control if your extracts are better or not.
Yes, we use it for the comparison purposes with our extracts, of course.
But this extract, it is obtained with the same procedure of your extract?
Well, it is very close, because according to the instructions, or we say the ingredients that were placed on the let's say on the box of krategut, it is also an ethanolic extract that is used for preparation of krategut.
Yes, yeah, because I want to understand this point, to make a good comparison between a standard drug that is obtaining with the same procedure for your extraction, ethanol, and yeah, and so on. Thank you.
Yeah. Thank you so much.
Not at all. Is there any question for the assistant? No question. I think it's clear. Sorry.
I want a small comment. Najat is my PhD student. I'm really happy to be advisor of Najat. I'm proud of you.
She's lucky. And she is lucky.
I must thank you for all the support you give me, for all the happiness you give me. Really.
Thank you
You
so
are
much.
lucky. Really, you are lucky.
I'm very lucky. I'm very lucky.
Yes.
Good luck. And
Thank
now, not at all. And now, please, Phyllis, to present the last presentation.
Yes. The title of the speech is Bioactive Compounds, Antioxidant and ACE Inhibitory Activities of Hippophaea rhamnoides and In Vitro Bioaccessibility Studies. I think that Eda Sensu will present this work.
Yes. Mine Gültekin Özgüven will present it.
Okay, sorry, Mine. We are listening you, Mine. Welcome to our session as a last speaker.
Thank you very much, Filiz Hocam. I want to share my screen first. I hope you can see. First, good afternoon. I wish you a very healthy days, especially in these hard days. I want to talk about bioactive compounds, antioxidant and AC inhibitory activities of sea buckthorn plants and in vitro bioaccessibility studies. In fact, this study is a part of a master thesis prepared by Eda Şensu, Miss Eda Şensu. And I am Dr. Mine Gültekin Özgüven from Istanbul Technical University, Department of Food Engineering. I will try to present her master thesis. First, sea buckthorn. This plant belongs to the Elaeagnaceae family, and it's widely grown in Central Asia, China, Siberia, North and East Anatolian region of Turkey, and it's locally known as yalancı iğne or karga dikini. And in fact, its medicinal use is available in the ancient Tibetan medicinal text. Medicinal text. In fact, the plant has very valuable berries, twigs or branches, and leaves. And we know that it's been traditionally used for the treatment of asthma, skin diseases, gastric ulcers, and lung disorders due to having very wide spectrum of pharmacological effects. So these are the commercial products available in the markets, such as jam, fruit juice, moisturizing cream, seed oil, tea, vitamin supplement, dietary supplement. And these are this is the list of these pharmacological activities of the different parts of the sea buckthorn. So you can see it has antioxidant effect, immunomodulatory effect, or hepatoprotective effect, or healing activity, and so on. It's a long list, in fact. And sea buckthorn berries, you can see the picture of the berries. The color may be range from the yellow, orange to red. It contains vitamins, particularly vitamin C and E, organic acids, tocopherols, phytosterols, phenolic compounds, PUFAs, and carotenoids. Its leaves are really valuable also. It also contains flavonoids, fatty acids, isoflavones, carotenoids, and proteins. However, these phytochemical content may change depending on the harvesting season, genetic variation, or climate, or post-harvest treatment. It may change. The majority of the uses in humans are based on the historical reports. There are only a few modern clinical investigations, and they are about on efficacy of seed oil for gastric ulcer, or improved atopic dermatitis, or affecting the risk factors for coronary heart disease, decreasing cholesterol level, and of course cardiac function, and also positive effects on other metabolic diseases. I want to give you an example. For example, the seed oil contains EPA, alpha-linolenic acid, linoleic acid, so it helps to improve atopic dermatitis, for example. So the seed oil is very also valuable. Regarding the cardiac function, we thought that if it had effect on antihypertensive activity, which is related to ACE inhibition activity. In the literature, there is very limited study, so we wanted to check this. Why is ACE inhibition activity is important? Because this is the pathway of the regulating blood pressure. So if angiotensin is converted to angiotensin 1, and it is also converted to angiotensin 2 by ACE, it's a kind of enzyme, then this angiotensin 2 affects the hypertension negatively. So we want to inhibit this ACE enzyme. So we also wanted to check this. We also wanted to check this activity. Safety, because if you want to eat the leaves, you need to know the safety. Again, there is very limited study in the literature, but hopefully no adverse effects of leaf, adverse effects of leaf aqueous extract were observed at a dose of 100 milligram per kilogram body weight per day in rats, administered for 90 days. And also no adverse effects in any of the groups were observed, administered with seed oil. We obtained, provided the plant from Bayburt province of Turkey, and we separated the fruits, leaves, and twigs, the parts, and we checked the bioactive compounds, including polyphenols and carotenoids, and we determined the antioxidant and ACE inhibition activity, and we also determined the in vitro bioaccessibility of bioactives of the fruits. We employed two different extraction procedures to obtain phenolic and carotenoid extracts, and we checked the total phenolic and total carotenoids and total anthocyanins by spectrophotometric methods, and also we determined the individual phenolics, and we are still working on individual carotenoids by HPLC and LCMS. We determined antioxidant activity by two different assays, namely DPPH and CUPRAC, and also we checked ACE inhibition activity. And finally, we use simulated digestion procedure to for analysis in vitro bioaccessibility. Now the results. When we check the results, let's start from the total phenolics. The green one, the green histogram belongs to total phenolics. We saw that the leaves was the first one. It contained the highest amounts of total phenolics, followed by branch or twig and fruits. And when we check the literature, our results are very agreement in the literature. You can see here, there is a really very wide range, in fact. And when we check also the Turkish genotypes in the literature, we can see our data is really close to the report. Also, when we check the anthocyanins, total anthocyanins, of course there is a very little amount of anthocyanins in berries. However, in the literature you can see it's very trace amount, but in our sample we got higher results for total anthocyanins. And when we check the total carotenoids, also we can find a very compatible result according to the literature. It's also very compatible with the literature. And when we check the antioxidant activity by DPPH and Kuprak assays, we again saw that leaf sample again exhibited the strongest activity among the parts, again followed by branches and fruits. And these results are also very compatible with the literature. So we can say that total phenolics of the berries are 4 to 5 times lower than red berries and equivalent to yellow berries. And nearly 2 times higher total phenolics was observed in leaves than many plant leaves. And finally, sea buckthorn total carotenoid content was similar to that in other plant foods, particularly rich in carotenoids, such as tomatoes, carrot, or spinach, or kale. And we also checked the correlation between the total phenolics and antioxidant activity. So you can see we got a very high correlation between them. Individual phenolics, especially catechins and epigallocatechin gallates, twigs and leaf parts are very good source of the catechins. And also these results are very compatible with the literature. Now, ACE inhibition activity, we wanted to check this because it's very limited in the literature. And again, we saw that leaf part exhibited the highest activity. But when we checked the correlation between ACE inhibition activity and antioxidant activity or total phenolics, we couldn't find a correlation. In fact, in the literature, some says there is a correlation between ACE inhibition activity and antioxidant capacity and also total phenolics, but some says there is no relationship between them. So we also couldn't find a relation, but we think that because leaves contain very considerable proteins, maybe this activity may be related to these proteins. Finally, we are simulating the gastrointestinal tract conditions. We are trying to make mouth, stomach, and intestines. We are adjusting the pH, body temperature, and also we add some digestion enzymes, bile salts, and other things. And at the end of the procedure, we got 2 fractions, which is one is in fraction, the other one is out fraction. In fraction, it represents the material that enters the serum, and the other one, the out fraction, represents the material that remained in the gastrointestinal tract. So, as expected, out fraction always contains more phenolics or more anthocyanins or more antioxidant compounds. It's an expected thing. We should focus on the in material, in fraction here. So we can see, regarding, of course, the literature, we can see relatively a very good recovery of bioactive compounds, total phenolics, total anthocyanins, and antioxidant activity regarding our berries. Finally, we can say that sea buckthorn parts very valuable. In particular, leaves, we can say leaves had the highest compounds, antioxidant activity, AC inhibition activity, and bioaccessible compounds percentages is really high, and we can say that we can use these fruit parts safely in food, cosmetic, and pharmaceutical products. Thank you for listening and attention.
Thank you so much, Dr. Mine, for this excellent speech. And many greetings to the Istanbul Technical University colleagues. Is there any question? I think everyone is a little bit tired, and we need to drink a I think my dear Nazım will thank us because we completed on time.
Thank you.
Thank you for all speakers, and thank you so much.
I wish if we were able to meet face to face to meet these beautiful faces.
Hopefully, dear.
We will do it again, inshallah.
Yeah, inshallah, next year or very soon. And now I'm closing this session, and we will start the next session normally 4:20, but we can start earlier. We can start earlier if we have some people from Peru. Peru, we have participants. Peru, it's very early in Peru. I think we can start 4:20 on time. On time.
Thank you.
Thank you
Professor
for your
Nazım.
participation.
Thank you very much.
Thank you very much.
Okay.
See you all
Also,
in next year face-to-face meeting, I think.
Also now Peru people came. we mentioned Peru people came, so we can start earlier, around 4 o'clock.
Okay.
4 o'clock we can start.
Okay.
Okay.
Have a nice coffee drink.
Thank you
See
very
you.
much.
Bye.
Thank you. Bye.
Okay.
See you, everybody.
Bye. أنا وجدي واحد
أمريكا؟ لا ما يشبه أمريكا.
شيرة أمريكا وولها.
ولها.
Hallo, ja. Okay, قاعد بيني دي تاكسي في لك.
Hello, everybody. Can you hear me? Good afternoon from Turkey and good morning, Peru.
Good morning.
And is there Elizabeth Paytan, Shalin, Professor Shalin is here? Elizabeth Paytan, Shalin, Professor Shalin is here?
Yes.
Professor Shalin, your name is Win Tan. You are Professor Shalin?
And good afternoon, doctor. Good afternoon. Excuse
Good
me.
afternoon. Professor Shalin is you. Okay.
Okay, see, I don't understand. Repeat, please. I don't listen.
Yeah. Okay. Welcome to MESMAP Symposium. Good morning to you. I think in Peru, how much the time? In the morning?
See, in the morning. I think that is in the afternoon. I think in the morning in Peru. See.
Okay.
Yes.
We are coming to evening here, and in China and Japan is sleep, preparing to sleep. Now,
Excuse me.
they were in the morning in our time. Now the session you are chair. You will manage. Also, I think Elizabeth Paytan is here. I think.
Okay.
I think she is Elizabeth Paytan. I think.
And she is in a present.
In present, I think.
In the audiophone.
Good afternoon.
Good morning.
Yes, good afternoon. Hello.
Yes. I think I changed your name. It's okay.
My name is Elizabeth Paytan.
Elizabeth, I changed your name. I'm sorry. Here, your screen, there was no name. I changed now. Elizabeth Paytan, welcome. And now we will start our presentation. You have 10 minutes presentation. Now your chair is Professor Shalin. You can introduce Professor Shalin to Elizabeth and invite.
Okay. Yes. Okay.
Professor Elizabeth, you can share your screen with us. The first, open your presentation on your computer.
Okay. Presentation. Moment. I cannot. 1 moment, please. Excuse me.
I can help you. The first, open your presentation.
1 moment, because I.
Okay.
1 moment, please.
Okay, we are here. No problem. It's now that it's shining. Sun is shining.
Yes. 1 moment.
If you want, we can start, Professor Shalin, and you can prepare your presentation.
Yes, please.
Okay, Professor Shalin, are you ready?
Okay.
Okay.
You
No
can,
problem.
you can, You can share your presentation, please.
Okay. No problem.
Okay, thank you.
You can see, please.
It's coming. Please full screen. Full screen at the first present.
Sí, okay. Excuse me.
Yeah. I can stop and you can stop.
Sí, un momento.
Okay.
Ay, salió.
Now.
Yes, can you see?
Now, okay.
Can you see? Thanks. Good.
You have 10 minutes.
Okay, thanks. This research is the title Chemical Composition and Antibacterial Activity of the Essential Oil of Salvia sagittata Ruiz Paz. This is the University Nacional del Centro del Perú. The introduction. The problem in this is, it is estimated that Peru has about 25,000 species of plants and 35% of which are endemic. These plants are in infusions, inflammatory, antiseptic, antipasmodic. No? Entonces, these plants have Salvia sagittata, and this study so that its components. The essential oils are aromatic. Essential oils are liquid viscose aromatic obtained from plants, no? By different extract for how oils. It is used in the pharmaceutical industry, in the cosmetic industry, in the industry food. This plant, this plant sagittata, it's flower blue. Objectives: evaluate the chemical composition and antibacterial activity of the essential oil of Salvia sagittata Ruiz Paz in fresh and dry stage against Escherichia coli and Staphylococcus aureus. Materials and methods. This research plays of execution on the laboratory microbiology of the University of Center of Peru and Natural History Museum of the National University of San Marcos and Lavicer Laboratory of the University of the National University of Engineering. Vegetable material. It is Salvia sagittata, fresh and dry stage. Salvia sagittata origin in the town of Yerba Buena, Guasaguasi district in Tarma, provincia in the Junín department. No? It is material, it's an altitude of 27,000 in this Peru. Biological material, Staphylococcus aureus, ATCC, 1923 and Escherichia coli ATCC. Antibiotics, it use oxacillin, ampicillin, reagents and culture media, Trypticase soya agar, Trypticase soya broad, Muller-Hinto agar, Muller-Hinto broad, and Mexer-Farland comparison standard. Laboratory. In this research, the gas chromatography coupled to the mass spectrophotometer in the National University Engineering. And the other materials and the reagent. Experiment methodology. In this research, raw material, it is taxonomic classification, Salvia sagittata. It's the method of Cronquist classification system. Then selecting and drying, extraction of essential oil by steam extraction. This essential oil physicochemical analysis and determinants of compounds. Then determinants of antimicrobial activity against from Staphylococcus aureus, Escherichia coli. In this photograph, collect oral material, la salvia sagittata, extraction of essential oil by steam stripping method, no? And this vegetable salvia could with approximate 6 centimeter to 8 centimeter with 20 kilograms of salvia was placed with 20 liters of water place. It was heated until the evolution of water vapor. It was decanted. The oil was deposited in hermetical sleeve dark glass flash to analyze. This photography, no, the evidence of research. Determination of chemical composition, and in the components were identified by comparing the mass spectra of the peaks of the chromatin of the sample with the mass spectral of the standard reference data of the National Institute of Standard and Technology. And this research determination physicochemical characteristic, relative density, relative index, acidity index, and yield. Determination of sensitivity, no? The method of Kirk-Bauer. And this percentage of exhibition essential on exhibition halo and control exhibition halo for 1%. Determination of the minimum inhibitory concentration is the method of microdilution that equipped use turbidity spectrophotometer. Results and discussion. The result in this research, one, the taxonomy classification of salvia. Salvia, the scientific name was Salvia sagittata Ruiz Pab. Characteristics of the essential oil. Salvia in fresh, salvia in dry stage. In respect, the gel in not difference in the gel. And density, the difference, not difference, and 1.949, excuse me, density is not a difference. And refractive index, the salvia in dry states is more, no? And in index acidic index in the salvia in dry state is more and the salvia in fresh state. In the difference is that because the salvia is in different stage. Characteristics of the essential oil. Well, chemical composition of in essential oil of Salvia sagittata and 40 components. However, essential oil of Salvia sagittata in dry stage have 42 components. The components major is caryophyllene. No? In the Salvia sagittata and fresh have 58. In the Salvia sagittata in dry stage, caryophyllene 58 is different, is major in dry stage. Is part of chromaton evidence the compounds of the Salvia sagittata in fresh is the chromatogram in the salvia dry is different, is major, and respect the determination of sensitivity. The determination of sensitivity against Escherichia coli ATCC 25,922 y Staphylococcus aureus ATCC of Salvia sagittata, the essential oils, it's in this table. We see the in fresh stage is the halos mid-meter is 8, no? Y en dry is 8. Entonces, interpretation standard of diameter son ampicillin control It's essential oil is resistant and friend against sensitive Escherichia coli. And sensitivity of the Staphylococcus aureus in fresh stage and dry stage, essential oil is resistant too. No, the allos meter is 8, is 10, is low. And respect determining the inhibition percent and minimum inhibitory concentration of Salvia sagittata essential oil, it is Escherichia coli, su percent of exhibition is 27.25. In the Salvia in dry is 27.27. It's the same significativamente. Well, Staphylococcus aureus is more, it's 55.67, and in dry Salvia is 72.09, and it's different in the Salvia in dry is more. Is the determination exhibition and in percentage in inhibition against Escherichia coli is the same. Percentage in inhibition against Staphylococcus different. In the dry Salvia is major. Determination of minimum inhibitory concentration, el MIC, the same MIC, in the same bacteriums is 23.72 in the concentration 50%, no, in the same. And it is the research get this result. Is the evidence the MIC and Staphylococcus aureus the same again the same how is the Escherichia coli. Conclusions. Salvia sagittata Ruiz essential oils has adequate physiochemical characteristics. The main component identified was caryophyllene and other secondary metabolites. The bacterial strain of Escherichia coli was found to resistant to the antibacterial action of Salvia essential oil, fresh and dry, with an inhibition percent of 27.25% and 27.27 respectively. The bacterial strain Staphylococcus aureus turned out to resist to the antibacterial action of Salvia essential oil in the fresh and the dry stage, with an inhibition percent 59.67% and 72.09% respectively. The minimum inhibitory concentration of the Salvia sagittata essential oil in fresh and dry stage against Escherichia coli and Staphylococcus aureus at a wavelength 129 nanometer at a 50% concentration. Thank you, National University of the Centro of Peru. Thanks. And any question, please? I hope I understand you. Excuse me.
Thank you, Professor Shalim. Is there any question to Professor Shalim? I think there is no question. Professor Elizabeth, are you ready? Professor Elizabeth.
Yes.
If you are ready, we can attend with you. Otherwise,
yes. Okay. Good afternoon, everyone. Thank you to the organizer for giving me this opportunity. In my country, there are not statistics data on blackberry production, and it is only found on the Y. These fruits are produced in the district of Tintaypunco in the brine, in the provinces of Tayacaja, Huancavelica, Peru, as well as the upper parts of Lima and the other parts of the central region of country. But blackberry fruits are not consumed by the population. In some case, it is used to treatment some respiratory ailments. Most of this fruit is for beer consumption or is not to the hurt or harm when the people consume. This is due to ignorance of this composition and the species to the which it belongs. And in this context, the aim was to the characterizers and the determinate phenolic compost in the concentrate pool and without concentrating in two mature stages of the rubus ECPP wild fruit from Tintaypunco. For taxonomy, it was done according to Cronquist and his parents' group. The qualitative characteristics was made by morphological descriptors according to the range. The qualitative characteristics show the weight and the length for the samran. Physicochemical analysis, acidity by Colombian normal techniques. pH, degrees Briggs, acidity, titular acidity, protein, fat, humidity as crude fiber, and vitamin C by AOC method, and carbohydrates by difference. Total monomerics and tocenin by difference pH method, the France, and modified or used analogs. The total phenolicus by singleton and antioxidant capacity by Branch Williams. Our team collected leaves, flowers, fruits, and branches. We took in the biology laboratory on the Agrarian University. To pool concentration, the pool, the rubus ECP, when collected, selected pulpits and concentrated to 50 grade C, up to 20 grades Briggs and bushy rotary evaporator, and constant pressures of 400 millibars in the pool was the almacenation for freezing at minus 20 grades C, where they were used to the correspondence for the frost. The results in phenotypical taxonomy determination, the examination and recognition of the morphological characteristics of the quality and quantitative orders in the sample allow conditions that it corresponds to the species Rubus stachifolius poi. These are the A's of the leaves, and these are all the leaves. The flowers are white, and these are the fruits. Phenotypic taxonomy determination of the wild blackberry according to Cronquist and the ancestral phylogeny is order Rosales, family Rosaceae, genera Rubus L, species Rubus urticifolius poi. The fruits have many drupes, and these are the physiologically mature fruits, and these are mature fruits. And they have a grade 6 and grade 3. Physiological characteristics of blackberry fruits, physiologically mature and physiologically mature and mature, and the weight, diameter, and left in the mature fruits were higher than the physiological mature. A physical chemical analysis of fruit pulp without concentrated pH, bricks, and reducing sugar in mature pulp were higher than physiological mature, but moisture and acidity were lower. Proximal analysis of the concentrated pulp to blackberry. The concentrated pulp, there are significant differences in ags, fiber, and protein, but moisture, fat, and carbohydrates, there are not. Vitamin C in pulp with blackberry. The concentrated and without concentrated mature pulp had lower vitamin C than physiological mature pulp. The pulp concentration temperature affects the vitamin C content. If there is more temperature, the lowest vitamin C. Total monomeric anthocyanin content in pulp blackberry. The mature pulp has more monomeric anthocyanin than physiological mature, and concentrated has higher increase. Polyphenol contained in the white blackberry pulp. The concentrated and without concentrated mature pulp had more phenolic compounds than the physiological mature. The concentration increased more the compounds. In this graphic, the concentrated and without concentrated mature pulp have more antioxidant activity than physiological mature. And concentrated mature pulp in comparison with without concentrated has higher increase in antioxidant activity because there are more phenolic compounds. In conclusion, the phenolics that wild blackberry correspond to species Rubus pistilifolius spoil. The total phenolics compost in the pulp 252,952 and 173,903. Finally, significant differences in no concentrated and concentrated pulp. 672 and 395,810. The concentrated mature pulp had higher antioxidant activity than phenolic mature. And thank you very much for your attention. I see there are no questions.
Thank you, Professor Elizabeth. Your chair is Professor Shalim. Is there a question?
No question, Doctor.
Recently, there are many wild berries or berries freeze-dried, and in the market we can find dried with original color and taste. I think their antioxidant activity, also phenolic content, are the same as the fresh ones or not. So freeze drying is a new method for drying to fruit. Do you have any study?
Yes, there are a variation, but in my country there are not there are not in the market, there are not this pulp. And in now the university, I am making this product because in the market there are not this product.
Recently in Turkey and Europe, especially in Eastern Far East countries, and dried ginger, dried mango, dried berries, and many in Turkish market is very popular. And I believe that they keep their original content. And thank you, Professor Shalin. Your second presentation, you can.
Okay, well, I screen. Can you screen, please?
Full screen, please.
Can you screen? Can you screen?
No,
can
okay,
you
okay now.
Okay. Good. The second research is chemical composition and antibacterial activity of Pachamuña essential oil, Edeoma mandoniana wet, extracted by supercritical fluids. The new methods different and the other research. Well, in introduction, a variety of plants native of Central South America, more especially to the Cordillera Andina, which are known locally as muña, have a characteristic mild odor. How to plant Pachamuña on the main family from the high wells and mountains. No, it's the family muña, the Pachamuña. It is made to Peru. The essential oils are semi-volatile and aromatic viscous liquid substance obtained from plant material extracted by different methods, such as supercritical fluids. The method supercritical fluids extraction throws is an interesting alternative to obtain, purify, and extract very specific compounds without using toxic compounds. It is used in the same pharmaceutical industry, cosmetic industry, phytosanitary industry, and the use in the industry food as natural preservatives. Objectives. Evaluate the chemical composition and antibacterial activity of the essential oil of Pachamuña, Edeoma mandoniana wet, against Escherichia coli and Staphylococcus aureus, obtained by the supercritical fluid method. The same place of execution in the University National in the National University of the Center of Peru in Peru, and the National History Museum of the National University of San Marcos, La Visser Laboratory of the Faculty of the Science of the National University of Engineering. The vegetable material is Pachamuña in dry stage. Origin, Pachamuña was collected in town of San Pedro of Quisquar in Tarma province in the Junín department. It's in the all altur. Biological, and the same Staphylococcus aureus, Escherichia coli, the antibiotics oxacillin, ampicillin, reagents, Triptychus soy agar, Triptychus soy broth, Muller-Hilton broth. In the same gas chromatography, it is used chromatography coupled to the mass spectrophotometer of the National University Engineering. Experimental methodology, it is raw material, taxonomic classification, Edeoma mandoniana wet. This picture is the plant, selection and dry, humidity, extraction of essential oil by supercritical fluids. And that's physicochemical analysis and determination of components. In the other, determination of antimicrobial activity Staphylococcus and Escherichia coli. Extraction of essential oil by supercritical as a solvent, and it is Pachamama, selection, drying, temperature 1820 Celsius, grinding, diameter 0.3 and 0.5 centimeter, and then 7 diameter diameter 429 micrometer. Oil extraction with CO2 solvent. This parameter is 150 grams of Pachamama, temperature 40 Celsius, pressure 150 bar, time 90 minutes. And then oil collection, then analyze. This picture is the equipe of the supercritical fluid. In the laboratory, the faculty industry food. Okay, the determination of chemical composition is the same the other in research. The components were identified for comparing the mass spectra of the peaks of the chromatogram of the sample with the mass spectral of the standard reference data. No? Gas chromatography and mass spectrophotometry. In this research, determination of physicochemical characteristics, relative density, relative index, and yield. In this research, to determination of sensitivity to the method of Kerber-Bauer. No, this bacterium dilution distilled water inoculum. It's to isop in the essential oil with plant disc, obtain a result. The results percent of inhibition is equal essential inhibition in allo, enter control inhibition allo, percent 100. In result and discussions. This research, taxonomic classification of Pachamama, order, family, specific, Edeoma mandoniana web. It's identified in the taxonomic classification. And vulgar name, Pachamama. Characteristics of Pachamama, essential oil in dry stage. On dry stage in this research. Well, characteristics, gel, percent, no, 2%. It's more high. It's more high in the other studies of essential oil. Density 0.976. It's different, it's major. Refracting index is 1.98. No, it's different. It's more characteristics of the essential oil in Pachamama. Chemical composition of Pachamama essential oil in dry stage. This compound is pulegenome. No, is 60.95 is old. Other components obtain acetato, linalol, cubenene, limonene, pinene. It is essential oil have 24 compounds. It's their part chromatogram, this invention of the compounds, this oil essential. Determination of sensitive. In the determination of sensitive against Escherichia coli, Escherichia coli and Staphylococcus aureus, this first sensitivity of Escherichia coli, the allo is 60.43 meter, no? Interpretive is intermedio, sensibility is intermedio. And against Staphylococcus aureus, it is major, no? Is sensitivity major 13. This is the allo against Staphylococcus aureus 17.08. No, is major, is high. Determination inhibition percent in essential oil. Determination of the percent inhibition and medium of the essential oil Pachamuña, gram para Escherichia coli es al halo diameter is 60.43 and percent of inhibition is 53.21. No, it's more than the other plants. And against Staphylococcus aureus, halo diameters is 16.08 and the percent on inhibition is 121.08. In the future, see percentage of inhibition, the difference and against Staphylococcus aureus is more. Conclusions. The Pachamuña Idioma mandoniana essential oil has adequate physicochemical characteristics. It has been 24 compounds were identified. The component with the highest percentage was pulhona. These essential oils were previously described with antimicrobial activity. The antimicrobial activity tenfold as the essential oil of Pachamuña has a sensitive activity of Staphylococcus aureus and intermediate activity on Escherichia coli, which could indicate that essential oil can be used in the development to the products that require control of Staphylococcus aureus. Thank you very much. Any question, please?
Yes, I have a question. Shalin, thank you very much for your good explanation about the Pachamuna volatile oil. I have a question about the obtaining method of the volatile oils. You know that we use distillation methods for obtaining volatile oil. If the aromatic plant contains more than 1% volatile oil. I think Pachamuna yields 2% volatile oil. Why did you choose this supercritical carbon dioxide extraction method instead of the distillation? You know that supercritical carbon dioxide extraction method is generally used to obtain fatty oil and the other secondary metabolites and primary metabolites of the plants. Why did you choose the supercritical extraction method to obtain the volatile oil? I really want to know.
Okay, I understand very well. The method of supercritical fluids is high cost. It's different and is steam or the other methods, no? And its cost is in but its method of fluid supercritical, the components was identified more in the purity oil. Essential oil is purity is major in characteristic of chemical. Is the cost is high, no? Is high and is difficult in the research in this plant in obtaining essential oil, no? I understand. Do you understand? I explain very well.
Sorry, I didn't understand clearly. Distillation method is cheaper than the supercritical carbon dioxide extraction method. You need some special instrument for the supercritical carbon dioxide extraction method. And 2% volatile oil containing plants.
The other method.
I really want to know, why did you select, why did you prefer to use supercritical carbon dioxide extraction method instead of the distillation?
Of course, yes. Yes, the supercritical fluids is measured and the distillation, but the cost is high, no? But the components identified more is high, is purity, the yield is high, and the other methods. And the problem in the supercritical fluids is the cost. No, but for the other identified high components, purity, major yield is major, no? Okay.
During the supercritical carbon dioxide extractions, you may obtain the other compounds, for example, fatty acid, besides the volatile oil. It will be a kind of purity for the volatile oil. What do you think? Okay, I will write later and we will discuss together. Okay, thank you so much.
Okay.
Many greetings to Peru.
Oh, thank you very much.
You're welcome.
Okay. I just have a quick question for you.
Okay.
Yeah, could you tell us quickly, what was the percentage difference in activity, minimum inhibitory activity of Pachymonia against these two microbes compared to the antibiotics, ampicillin and oxacillin?
And you understand well. Excuse me, repeat, please.
You compared the minimum inhibitory activity of Pachymonia on two microbes, E. coli and the other one, the Salmonella, I think. Yeah.
Yeah. Okay.
So when you compare that with the standard antibiotics, ampicillin and oxacillin, what was the difference?
Okay,
which one was more, which one was better in inhibiting these microbes? The antibiotics or your Pachymonia extract? What was the difference?
Okay. Essential oil Pachymonia in the Staphylococcus aureus is more sensitive, and the Escherichia coli because is gram-positive Staphylococcus aureus, and Escherichia coli is gram-negative, is more resistant, is more the compounds of essential oils attach the parent, the wall, the cell in the Staphylococcus aureus, and in the Escherichia coli doesn't stay in the wall the bacterium. No, is the difference for the gram-positive Staphylococcus aureus and Escherichia coli is gram-negative. Is more toxicity is more dangerous in the Staphylococcus aureus, no? Is gram negative Escherichia coli is more resistant in the wall, the bacterium.
Yeah, so that is resistance related to the antibiotic, right? My question was, how does your extract compare with the two antibiotics?
Excuse me, repeat please.
How does the your Pachamuna extract compare with the two antibiotics in killing either the gram negative or the positive bacteria?
Well, the Pachamuna oil essential oils is the compounds pulegenona, no? It is monoterpene is dangerous to the gram positive and against gram negative. It's understand well.
Yeah. Okay. The minimum inhibitory percentage for ampicillin was 53, and for oxacillin was 121. What were the relative values for the Pachamuna essential oil?
Okay, bien. Essential oil of Pachamuna is the compounds 24, and pulegenona, linelol, caryophyllene is other compounds, monoterpenes, diterpenes.
No, not the composition. I was asking for the minimum inhibitory concentration or the minimum inhibitory percentage.
Okay, concentration. I don't time to concentration. Only researches the percent inhibition and the allometer. No, in this research, only percentage inhibition.
Yeah. So how much was that? Percentage inhibition.
Claro, percent inhibition. Bueno, for the Escherichia coli 23rd and Staphylococcus aureus 150, 51, 50.
That's pretty high. Okay. Yeah, got that. Thank you. Very good presentation.
Okay. Thank you very much.
All right. Thanks.
انت ما كيش فاهمه زعما انا شاف ديكيب وديري وهذا اخر شاف ديكيب ما كيش فاهمه هذا هو مالكي
Someone's microphone is on. Yeah, while we are waiting, I once visited Peru. We had a medicinal plant conference in Peru.
Okay, thank you very much.
Surely we should visit Peru together.
Yeah.
Together.
Miraflores, yeah.
Okay. I think that now we can visit Brazil. Dr. Juliana Cristina.
Okay.
Hello.
There? Yes.
Do you accept us to Brazil? We start from Japan and coming to Peru and Brazil.
Wow.
in the early morning we start, and now we are in after Peru. Now we are Brazil.
Okay.
Across the world.
Yeah. It's thank you very much. Please share your screen.
Okay.
full screen. Okay, now. I will stop.
Okay, okay.
Please share again.
Okay.
I think there is technical problem. Now, okay.
Okay. Oops.
My name is. Good evening. My name is Juliana Bastos, and I am a PhD student at Federal University of Ouro Preto, Minas Gerais, Brazil. And I would like to present you our work entitled Development of Nanoformulation Containing Whale Herb Oil. Cordia verbenaceae, commonly known as whale herb, is a shrub native plant to the Atlantic forest region, which has some biological activities described as anti-inflammatory, healing, analgesic, antifungal, diuretic, laxative, and muscle relaxant. Its leaves have a group of chemical compounds such as monoterpenes, tritamines, flavonoids, and fat acids. The main compounds are beta-caryophyllene, burlene, and elixir, which are related to their biological activities. Currently, there is a growing interest in working with medicinal plants to development of new formulations, among which nanoformulations stand out. These formulations can enhance the permeation and penetration power of these compounds, optimizing the therapeutic action. Based on this information, the objective of this study was to develop a nanoformulation containing Cordia verbenaceae oil. For that, the oil was extracted from the leaves using the hydrodistillation method. This photo shows the extraction process. After oil extraction, the nanoformulation was developed using the components described. Formulation A contains all components except polyethylene glycol and DMSO. In this formulation, Cordia verbenaceae oil was solubilized in hydrogenated castor oil. As you can see in formulation B, the Cordia verbenaceae oil was solubilized in polyethylene glycol. And in formulation C, the Cordia verbenaceae oil was solubilized in DMSO. The quantity of the components has not been demonstrated because the nanoformulation is under patent registration. And for the nanoformulation production, the aqueous and oil phases were heated separately to 73 degrees with a variation of 2 degrees and shaking at 400 RPM. After cooling to room temperature, the formulation was evaluated according to the parameters: microscopic stability after 24 hours, average diameter, polydispersity index, and zeta potential measurement. Visually, formulations A and C incorporate 100% of the oil and prove it to be stable. However, formulation B did not incorporate all the oil, causing separation of the nanoemulsion in two phases, as shown in the figure. The average diameter of formulation A was 181.6 nanometers. The zeta potential was -44.2, and the polydispersity index value of the formulation was less than 0.3, characterizing it as a non-dispersive system. Therefore, the formulation A was selected in our study because it is stable and is within the parameters of non-emulsion characteristics. alternative to enhance the therapeutic effects of whale herb oil. I would like to thank you for your attention, and I leave my email here for questions or suggestions for our work. Thank you very much.
Yesterday, but he was not available, so we changed the program. We write the name in today. I think I write the message, but yes.
Is there a question?
Okay.
Is there a question to Brazil? It's very interesting. Can you hear me, Juliana?
Hello?
Can you hear me?
No.
No?
No. Please
now,
wait a
can you hear
moment.
me? Can you hear me?
Okay.
Now?
Hello?
Hocam ben duyuyorum şunu.
Evet, duyuyorum.
Ha, Juliana duymuyor o zaman sadece.
Juliana.
Oi. Hello.
Hello.
Hello.
We can hear you. We can hear you.
Hello.
Juliana. We can hear you.
Hello.
Please use your cable. Your cable check.
Please open your earphone, maybe.
Yeah, please. Maybe.
Hello.
Juliana, remove your this. Can you hear?
Hello.
Yes, we can hear you. I think you cannot hear.
I think she has some
Yeah.
problems.
Some technical problems, I think. Juliana. So last year, or she can try again. Last year we have a presentation from Malaysia, and there was some plant names: elephant herb, cat herb, and different animal. I say in the first time in the title, and as to the professor, your presentation is related to medicinal plants. Are you sure? Not herb. There is cat, like different organs of the animals, like elephant ear and some lion hair, like that. It was interesting, and he sent to me Malaysian professor Mustafa Rais. He's very popular professor, and I say, Professor, this is medicinal plant symposium. Are you sure your title is related to medicinal plants? So it's not cat leg or elephant ear and lion hair, and this is some animal parts. He sent to me the original and Latin name. I say, I'm so sorry. I'm so sorry. I need to read more. I need to visit more countries. Up to now, 80 countries only.
I think that English name and local name of the medicinal plants can be different. So why we need the scientific name, Latin name of the plants?
We want to in another salon. There is two presentation. I think they will finish in 15 or 20 minutes, and we will suggest them to come to our salon online. Our salon is very big, you know. Guest room. Guest room. Salon A is guest room, and now everybody is awake. Maybe other salon, some people in Japan or Thailand may be sleepy. And now we are in South America, and we are active, and we can wait some 15 or 20 minutes. And Dr. Sevgi is very tired, and she say, okay, confirm me. And about 5:20, we can start closing ceremony. And normally we publish and release MESMAP-6 poster, our YouTube channel. All the participants can follow the posters of MESMAP, and also they can contact the authors of the posters. This time we cannot manage another poster session and live, but it's very good. We can use the YouTube channel from the MESMAP, and after the symposium, also you can watch and see, and you can take your photograph from the videos, MESMAP videos. And up to now, we are very happy. We are some little tired, but we are very happy. The first day, 20 invited talks, and today around in only Salon A more than 20, around 30 presentation, oral presentation, and like face-to-face meeting. And I don't want to talk more, so I summarize under like closing ceremony. I can listen you, Filiz Hocam, and you can share your, or you can drink tea or coffee. After that, we will post you your coffee and tea. Now you can use your coffee and your tea.
Thank you so much.
We are here, and please keep silent. In 15 minutes, we will start closing ceremony 5:30. We are here. Okay.
We should stay at this channel.
Yeah, in this salon. In this salon, we will invite Salon B. They will come to our salon. We will accept them. Now we are here.
Galiba birkaç kişiyiz.
Şu anda hocam
Şu
15
anda
kişiyiz.
kaç gibi ama
Şu anda 15.
kapalı
Orada
her-
kaç kişi var hocam? Evet. Orada da 15 varmış.
Mutlu hocam da var. Filiz hocam, hürmetler. Nazım hocam, Nazlı hocam.
Merhaba, merhaba.
Merhabalar, merhaba. Pınar hocam nasılsınız?
Nasılsınız?
İyiyiz siz?
Teşekkürler. Böyle bir ortamda sizlerle buluşmak tekrar çok güzel.
Aynen, maşallah Nazlı hocamın enerjisine hayranım. Yani ilk dakikalardan son anlara kadar böyle onun enerjisinden alıyoruz biz de. Şu anda bu arada YouTube'dan canlı yayındayız sayın hocalarım.
Öyle mi?
Neyse, yani herkes zaten şeyle Zoom'da ya. Yani çok ilginç böyle şeyler, anekdotlar oldu. Yani canlı yayın denemelerinde falan tabii şeyin azizliğine uğrayan arkadaşlar falan oldu. Böyle aslında şey oluyor ya, kamera arkası görüntüler oluyor ya. Onlarda bakın hala gelenler de var. Hoda Eğit geldi, Ali Bilgili hocam geldi.
Aslında bu online simpozyumlar bir bakıma da iyi oldu. Koronavirüs nedeniyle uzun süredir görüşemediklerimizle bir araya geldik.
Evet.
Sevgili Nazlıcığımla, sevgili Pınar'la çok keyifli. Türkçe konuşmak da güzel ama yabancı misafirlerimiz de var.
Ben de onu şey yapacağım. So we are sorry, we are talking in Turkish, and so nobody can fly, also domestic because of the COVID, and Professor Phyllis living in Cyprus Island, and other professor, other cities, and people miss each other. And this kind of meeting places is very good for talking each other, and so also new participants are coming, and oh, it was very good. This time hocam kaç tane kaldı? Son bir mi? Kim? Sana da çok çabuk kesin ya. Şurayı çok bitireyim.
At the end, Nazım left us. I'm he's running off.
I am here. I am arranging another meeting.
Okay.
It's the last presentation. I say, please hurry up.
Okay.
I am here.
Unfortunately, I couldn't attend all the presentations because I have some online lecture for the undergraduate students at Faculty of Pharmacy and Faculty of Nursing. So today in the afternoon, I can't attend all the presentation, and I'm very happy to see all of you.
Then you can watch us from YouTube.
Yes.
We are famous.
We are becoming famous. We are becoming famous.
Yeah. We have a big project on turmeric. Like 60 varieties, like they're green, violet, black, yellow, green, blue turmerics. They're all from Vietnam. So if anybody is interested in any collaborative research, particularly pharmacological, they can approach me and I can send the samples. We are doing one with a person in Nigeria and one in Sweden. So if somebody is interested in doing some work, let me know. We can provide some different types of turmeric and you can compare them with your local herbal plants. It will be an interesting collaboration.
No, it will be very good if you write us your email address, maybe.
Yeah, my email address is on the abstract.
Okay. I will check and I will write you.
Yeah. Okay.
Me too.
Yeah, thank you very much.
Yeah. You're
Thank
most
you.
Look forward to some collaborations.
Okay.
Yeah. Nazim, I have sent you the ACMAP flyer yesterday.
Yeah, I am so sorry I couldn't reply the emails. Only
No,
I can.
Yeah, but if you can publish
I
it
Yeah, please send me. I will share, and I send to all our participants. And so for 2 days, I can go only to home and sleep because it's too difficult to manage online.
That's right.
Face to face is really easy. Really, I miss the face to face symposium.
Lot of presentations. Yeah, that's good.
Next time we are waiting to in Izmir, Professor Şekeroğlu.
Yes. Next time, this time we couldn't manage in Ephesus. Ephesus is a great place. Hocam şeyi gönder, Hasna'yı buraya gönder. Burada konuşun Hasna. Ha, yapmaz. Şimdi bir tane sunum kalmıştı. O şu anda bitiyor mu hocam? Tamam, son sunum yapılıyor hocam. Yes, normally we arrange in Ephesus, but we are very sorry. So we organized a great hotel, Richmond Hotel. It was near the seaside and very good meeting salon and good activities near the ancient city of Ephesus and some religious places, very popular. And also we were waiting around 500 people. Normally we organize MESMAP symposium around 300 people, around 40, sometimes 50 different countries through the world. For Izmir, we were waiting, but this time we couldn't. We postponed the first. We wait to move the COVID from the world, but it insists I will be here. I will, but we move it by medicinal plants. I will make some spray. I will make some spray and we make first and go. go to other side the COVID. I say challenge. Who will remove from work? Me or COVID? We are sorry, last 6 minutes we will finish and we will start the closing ceremony. Now we are really happy when we start the MESMAP symposium in 2013 in Cyprus with my dear İlkay in pharmacy faculty, Professor İlkay. She was dean of the pharmacy faculty. She said we can entertain the people in Cyprus and a very good place, our symposium, and the university also supported us. And we start very good. And then people want to come, and again, we are very happy. This is the 6th edition. Some people say to us, many symposium we know. After the third symposium, they finalize. They cannot manage. I believe so. It was really difficult to manage, but we think amateur, not professional organization. So all the participants are our friends, our brothers and sisters. When we open the symposium, we say brother and sister, welcome. Also, when we organized before MESMAP a big congress, AMAPSEC Congress in 2010, we have participants from 53 different countries, around 500 or 600 people. It was really interesting, and now I can say we have many friends, brothers, sisters all over the world, and when we visit them, they entertain us. When they come, we entertain them in different parts of Turkey. Turkey is the middle of the world. everybody come easily. And Turkish Airlines is our sponsor, and we are happy. So they can fly Turkish Airlines before COVID and after COVID. More than 300 destinations. It's crazy. More than 300 destinations through the world. And Turkish Airlines is our sponsor after Barcelona. And our team is playing with Barcelona in the Super League.
Turkey has been known historically, right? So a lot of people from other countries are interested in Turkey. Turkey has been very active globally. Several civilizations there. Even with Indian civilization, we had interaction with Turkey. So the Muslims who live in India, they are still called Turki in our language. Turkey again comes from Turkey. People from Turkey.
Up to now, I visited 8 times India. And some people say one time I was in the market and some Indian people come to me and they say, which part of India you are? Okay, which part? You say? He said to me, you look like Kashmir. Yes, I say, I am from Kashmir. He believed. And in the evening, the cocktail, I said to some ladies around me, I said to them, some people asked to me, you are which part of India? And I say, Kashmir, he said to me. They said, Kashmirian guys are very neydi yakışıklı? Hocam yakışıklı neydi?
Handsome.
Handsome. The more handsome guys come from the Kashmir in India. So, so I say, oh, okay, I am from Kashmir.
I think Kashmiri people have Aryan influence. So Kashmiri girls and boys have European complexion.
Really?
Yeah, because they're higher elevation, they're in a cooler climate. And if you look at the
I'm lucky.
Kashmiri people, they're very fair and tall, and they have more European features. So we call them, there is a movie, they call it Kashmir Ki Kali, means the flower of Kashmir.
I can be artist in India. After being academician, I could be artist in Indian cinema.
Yeah. It's a nice movie if you watch Kashmir Ki Kali.
Yes. Hocam bütün şeyleri MESMAP'a gidiyorlar. 1 minute, I will manage other salon. 1 minute.
Okay.
Professor Mantredi?
Yes.
Yeah. You ask some collaborations for pharmacologic studies. That's only for some pharmaceutical approaches, for example, kinetics or toxicology studies. You mean that?
Any kind of collaborations, that what we are not doing here, we are welcome. My idea is to expand and see, get the maximum out of that crop.
Yeah.
So we have good variation in the varieties, so they will have different properties.
Yeah.
And we can get to see. We are doing antimicrobial properties, and I've been looking for someone to do anti-cancer.
Mm-hmm.
Yeah. and we will see how that works. And
Mm.
one group in Nigeria, they are looking at neurological properties
Mm.
because it is known to be active in Parkinson's disease and also Alzheimer's disease. So there may be some neurological effects. So they're looking for that. So
Yeah.
you can let me know what area you interested. Then shipping the
Yeah,
material
of course.
is not difficult. We have dried powder or fresh
Mhm.
rhizomes. We can ship them.
Okay.
And yeah.
Okay. We are working on some anti-cancer activities using some glioma cells, for example. So breast cancer cells, we are using them to check the anti-cancer activity of the several compounds, not only from plants.
So
I think we can collaborate in very soon because you know there is a big problem to combat the virus. Actually, we also able to do some antiviral tests, but not for this kind of viruses, of course. But soon, maybe I can find or I can arrange some laboratories to work together because you know this is very hard and this is I think it is the challenge for testing of antioxidant activity of natural products, especially for COVID-19 coronaviruses, because it needs very protected laboratories and several
that's true.
and professionals to deal with them. So here in Istanbul, even in Istanbul, we have some problems to do test the activities of natural products. There are some labs, but they prefer to work with some pure substances and known pharmaceuticals, currently known ones, known drugs.
Okay. Yeah, this turmeric is known for building up the immune system. So therefore, they are now consuming a lot of turmeric, ginger, and black pepper, a lot of tea made out of that decoction. And but that's only building the immune system, doesn't directly kill the virus. There's no evidence that it kills the virus. It just builds your immune system to fight the virus. Yeah.
Maybe it would be interesting to test their immunomodulatory activities.
Parce que جابوا كاع هنا
Prove it using the biological systems or using animal tests.
Yeah, that's not my area, but if some experts want to do it, yeah, I'd be happy to go with them. I hope the abstracts would be available, Dr. Sakaroglu.
I hope so. Next week, the first part of up to Wednesday. I hope so. So we publish abstract and full paper together in the proceeding book with ICBN numbers. So I hope so. Until at the end of next week, we will have finished the publishing. And I will give some information. People coming now, 27 people coming.
Yeah.
So we finished our team, Hasna. Hasna, thank you very much. Hasna only have 3 or 4 minutes presentation directly without any discussion. conclusion, and thank you. Thank you to Romania. This time we are very sorry. So people waiting, world is waiting us. Not we are now, I think more than 15 countries we are already here. From Romania, Italia, Peru, Brazil, America, Turkey, Northern Cyprus, Algeria. Hasna is our team. And
Thank you.
and so I think o taraftan mı? Yes, I want to give the first speech to Dr. Sevgi, associate professor, new associate professor. I will give to her the first speech, and then I will close the MESMAP-6 online symposium.
Before, I would like to say my sincerely thanks to all our participants from all over the world. Despite the COVID-19 pandemic, they are here and they support us. They send their valuable research and share their research data from our symposium. Hope to meet you all, our professors, our friends, our sisters, brothers, maybe next year face to face. Which city, I don't know, but we hope to meet you again like our previous symposiums. That was very nice meeting, I think. You shared valuable presentations. Thank you for your kind attention and contribution. Attendance certificates will send you via your emails. Maybe next week we will complete and send you. And some of the participants sent the full papers to us, and we manage and organize the abstract book and proceeding book. And in 10 days, we will announce from our website. You always contact us if you need any assistance or if you ask any question about meeting or abstract book and other things. I would like to thank again. Hope to meet you for the next one.
And,
I
yeah.
start with house.
Nazlı hocam buyurun. Nazlı hocam konuşmak. Thank you.
We thank you, Sevgi.
And thank you, Sergey. And now we have, I think, if there's waiting, Mihaela, we accept. And Sergey, do you have co? 1 minute. I can put Sergey to co-host, make co-host, and yes. Şeye bakarsın. Now, thank you, Sergey, and thank you very much for your kind participations to our symposium. Now, here we are 29 people, and we are happy. Normally, face-to-face symposium, sometimes we couldn't find the number. Only 15 or 20 people. Now online is sometimes maybe better, and we are very happy. We organized a 2-day symposium, and the first day, as you know, invited speech, and the second day oral presentations, and 2 salons. But in 2 salons we achieved, and I hope so. Everybody happy, and it's fruitful symposium, and our quality symposium quality is every year getting increase and increase, and now MESMAP is among the well-known symposium on the worldwide, especially medicinal and aromatic plants. It's very important to us. In 8 years, maybe for our Congress, total Congress for 10 years, we are organizing international symposium on medicinal plants, but only for MESMAP, 8 years we organize, and also we organized another symposium last year, also ICPBS, International Symposium Pharmaceutical and Biomedical Sciences. After this, Dr. Sevgi will be chair of that symposium. I'm retired. I am retiring little by little. I am so I am so tired. If I need any medicinal plants, give me big power, I will attend. Otherwise, I cannot attend more. So it's too difficult, and my hair is going white and white. So we are not lucky like ladies. Ladies can cover white ones, but Professor Rao and me cannot. So I have some hair now. I'm happy. Now we are closing the MESMAP symposium, and this year also we learned that the MESMAP symposium proceedings indexed by the Web of Science Congress Index. It's very good for us, and up to now we publish a special issue, very good journals. Also this time we will publish a special issue at Molecules journals. Participants will have the 40% discount at Molecules journals. But if you want to publish your full paper in the proceeding book, you cannot submit to Molecules. So many participants here published their papers in Molecules journals. And if you want, you can also send some Molecules level is very, very high. Sometimes it's too difficult. I am a guest editor. I couldn't publish my paper last year. It's very interesting. So we have our journal, Current Perspective Medicine and Aromatic Plants, CUPMAP journal, and now we have DOI number, and we are waiting to enter, indexed by the Web of Science in 2 years. Now we published 5 issue, and we will release the 6th one in this December. I hope so. Next year or another year, we are coming to Web of Science. So academically, I couldn't write a book, but with my team, we have good symposium, well-known and scientific, really scientific. In Turkey and around the world, many scientific, its name scientific, its name international, many symposium and conferences. Only some people entertain each other in their own language. So we start in English, and up to now, we didn't give any chance to give in any language, in both Turkish also. And people pray us, and many people in Turkey around the world and finish their PhD and they get associate professor degree, full professor degree by MESMAP symposium, and they pray, and we are here. I believe that. After this, also we are a big family, MESMAP family, and every time we are ready to help the people from far east, far west, far north, and far south and middle. We are here, middle. We are here whenever you need any participant, any speaker, any project partner, any people to help you without waiting any benefit from you. We are here. We are your brother, we are your sister in Turkey. Every time you are welcome in Turkey. Turkish hospitality is very popular. I hope when you come to Turkey, we can entertain you and we serve you kebab, baklava, and everything. I would like to thank you again and again, and I want to give you some chance to some professors if they want to say a few words now. I'm closing my speech and the MESMAP-6 online symposium. Thank you very much again. And if you want to say something, you can say.
Nazim, may I say something?
Please.
On behalf of the Romanian group, thank you very much once again. It's our sixth time participating in Turkish symposia and international Turkish symposia connected with MESMAP, including Yatus Pub and last year's pharmaceutical symposium. We felt very much supported by both you and Sevgi. And you mentioned the accomplishments you realize. It's a great symposium. We really missed very much this year the personal connection with everybody who participated. You have a great journal, the COPMAP, but you forgot to mention something. I think that the best part of this symposium is that it brought together people and gave young people a chance to collaborate and know each other. And the best example is Hasna and Kula, who are with us in an Erasmus program now with the Cluj University of Agricultural Sciences. And as you see, we took over from you this establishing connection mission, and we connected Hasna with our medical school in Cluj. So now they share publications. And I think that this is very, very important, broadening relationships between people, which, as I said again, I should mention it, we missed this year due to COVID very much because we enjoy coming to Turkey, and we missed the trip, which ends the conference, I must tell you, and also all-inclusive treatment from the Turkish side. That's always welcome, and we hope that we will be able to share this next year. So thank you once again for the great job you're both doing with the support of your university. And we hope that next year we'll be able to meet in person and continue this great tradition you established. Thank you very much once again.
I would like to thank the organizers of this wonderful MESMAP conference. You've been doing every year. It's a great job. I'm really surprised how you are able to manage to hold such a big conference year after year. And I see your program each year you send, and you have very good presentations, very good topics, and people from across the world. This is my first time presenting at this conference. I'm truly grateful to you and team for giving me this opportunity to present our research and meet so many and people from across the world. And like you said, it's like a global family, brothers, sisters, all of us. So I do wish we would continue to be in this kind of collaborations and continue to grow together as we understand better about these medicinal plants. Nature, we are fighting COVID, but when a nature gives a problem, it also gives a solution with it. Solution is just around us. It's a question of just seeing it and using that. So I truly believe that this MESMAP and several other, I'm with the American Council for Medicinally Active Plants, similar to MESMAP. We have also a journal of medicinally active plants. We do produce special issues after conference, and conference attendees have a chance to present full paper in the journal for no charge. So we will try to work with you how to amalgamate these two. You are in MESMAP is out there in Turkey, nearer to Europe, and we are here in US, nearer to the Americas. So maybe if we fuse together, we will have a much bigger global network. So look forward to that. And our conference is going to be in June, in person, hopefully. If government shuts down anything, then we will move to virtual platform. Otherwise, we are hoping to meet in person in Rutgers. And you all are welcome to participate. We are taking the abstracts now. Thank you so much, Nazim.
Thank you, Professor Rao. Also, Professor Randolph from UK.
Yeah.
Please, a few words.
I'd like to congratulate you again with organizing this very nice meeting. I very much enjoyed it. And also on behalf of the Phytochemical Society of Europe, I'd like to confirm that we will again support you in the next meeting and of course announce it to all our members through our websites. Like I said before, what I particularly liked was the breadth of the presentations, from full professors, highly technical papers to the young scientists. We all got equal opportunities to share our thoughts, and I think that is the key to good science, that there is a good discussion. So thank you very much.
Thank you very much. Also, we are waiting the next special issue journal from you. You don't promise, but you can help us about the special issue for the next MESMAP symposium, full papers.
Oh, welcome.
So your society, Phytochemical Society, support the MESMAP symposium. Hope so. We will have a very good Web of Science journal for next symposium as well. Now it's time to go. I think also people coming and again, and thank you very much again. Now we are closing, and if you have any question, you can contact by our email. But we say that we will send you your participation certificate and to your emails with barcode and signed, and with barcode you can QR code you can check your certificate is original or not, and also we will share further information our website mesmap.com and mesmap.org, and see you soon face to face. And we say also in Turkey, good evening. For Europe, have a nice have a nice day, and for South America, good morning. Now, good morning also. I think Charlene, Elizabeth, and thank you. We are very happy. 25 different countries and around 150 different participants, and around 60 oral presentation, 20 poster presentation, 20 invited talks, more than 100 real international presentation. We entertain you this time online, and hope to see you soon. And please take care of you and your family. In a healthy world, we will meet very soon. Thank you. I say bye-bye.
Thank you so much.
Thank
you so much.
Bye-bye.
Thank you. Bye-bye.
Nice organization.
Goodbye.
Thank you for everything.
Bye.
Bye,
Bye.
Diana. Bye.
Bye.
Bye, Sevki. Bye, Naz. Bye.
It is time to eat baklava. It is time to eat baklava because we repeated 5 times today. Bye.